Hello, everybody. Welcome. Great to see you all here. Thanks for showing up early, and for some people, and I guess late for others. I suppose I'll share my screen to start. So it is 8 o'clock Pacific time, and that is when it's supposed to start. I think we'll give people just a few minutes to catch up, and then I'll get started with the introductions of the conference, and we'll go ahead and get rolling around 13 minutes from now. Hey, good morning, Lori. Good to hear from you. Give it one more minute before we get started in earnest. I'm still waiting to see Christine online. All right, 8:03, time to go. Okay, guys, welcome to the third annual International Space Ecology Workshop. Um, it's a real pleasure to have you all here. I'm going to be the MC this year, where past years I was just in the technical support, but this year Christine Escobar has handed over the torch. Um, I'm really pleased to have everyone here. I'm really grateful to everyone who's volunteered their time and their expertise to come give us all of these really excellent talks. And of course, grateful to everyone in the audience for spending the time. This is kind of a long event to be sitting at your computer for 8 hours a day, for 2 days. Of course, this will all be recorded, so don't feel obligated to be here for every second of it. It will be available to watch on YouTube, probably within a week or so. Um, yeah, I mean, let's go ahead and get started, guys. Of course, this is the third iteration of this event, and we started it a few years ago because there's a there's no real home for us in the conference circuit, for us that are really interested in artificial closed loop ecosystems to support life, or all the other kind of broad spectrum of space life sciences and their interaction with the environment, which is what we call ecology, of course. Um, in in the context of this study, we're really interested in how we can use closed ecological systems to support life in space and also on Earth, especially in the face of more extreme climates. Um, and of course, how we can use these principles of closed loops to create circular economies and circular production systems to have a more sustainable way of living, both on Earth and in space. Of course, we're not the first people to think about this. People have been thinking about it for decades. It's just that in kind of the more recent decades, it hasn't been as popular. So this is our effort to reignite the conversation around closed ecological systems, space life sciences, bioastronautics is specifically a thing that we're including in the purview of this conference. And by bioastronautics, we really mean the human perspective on life in space. How do humans react? The call to action for today and for this event is we think that the best way to advance this as quickly as possible is to be welcoming to all of our colleagues in working towards collaboration in a cross-disciplinary way. And specifically, this is actually my little call out here is I really want to see an emphasis on multi-trophic approaches to the science we're trying to do in space. Because of course, we've done quite a lot of space biology, but it's pretty rare on Earth for biology to exist in a single species vacuum. So looking at the interactions between those species and their environment, I think is going to be really important to us getting a handle on how we're going to do support life in space going forward. So just as a reminder, if you guys weren't here for the very first Space Ecology Workshop, there's going to be a few differences between this one and last year and the year before that. In the previous years, we had a pretty big emphasis on doing we had collaborative whiteboards through Mural. We also had the breakout rooms. We're not actually doing those this year. Partially because it's pretty complicated to manage on the back end for me while I'm also being the MC, but also because we didn't get a ton of interaction on these in the past. We're definitely open to changing this in the future if there's a demand for it. So you guys can just let me know in the comment box. The chat log is actually something that will be saved and made available. So if you guys see information that you want to follow up on, or if you guys want to drop links to relevant studies, that's definitely a good place to put it. After the first Space Ecology Workshop, we did use all of these tools to survey the community and understand where we are and where we're going. We made a bit of a technology and knowledge gap assessment, and that is all available here in this paper that we published at the International Conference on Environmental Systems. This is open source, anyone can access it. There's a link here, but I think if you just Google that title, you'll be able to find it. It's also on the website, Space Ecology Workshop. Let me introduce myself, I guess, and my co-organizers. It's a slightly different lineup from past years. People had to come and go as they've been busy with other things. Of course, there's myself. I'm Patrick Grove. I am a co-founder of the Spring Institute for Forests on the Moon, which is a European nonprofit research and development organization. I guess I can just introduce everyone, huh? We've got Jane Chetsoff on here. She is a professor at UCLA, and she's also the president of the Propagule Space Ecology Institute, which is I'm also a part of this. It's a nonprofit here in the United States, which is the governing body for this conference. And also we're working towards the same thing everyone else is, working on fun bioregenerative life support system technologies. And specifically, one of the goals of Propagule is to address the needs that we identified in that paper I was mentioning. We're trying to direct funding and efforts for collaboratively progressing towards closed ecological systems and closed ecological life support. Also helping us organize this year is Marshall Porterfield. He's a professor at Purdue, former NASA director of biological physical sciences, and he helps us at Propagule Space Ecology Institute. We've got Frida Taub on here. She is a professor emerita at the University of Washington and an early pioneer of closed ecological system sciences. And of course, there's Stephen, who is I think he's still working on the PhD at University of Florida and is involved down there in NASA as well. All of these sponsoring organizations you see at the bottom are mostly just our affiliations. This this is a free conference. Nobody actually pays us to do it. So, yep, that's that. So we're going to go over the agenda really quick. Um, like past years, we're doing a mix of featured speakers, which are going to have a bit longer talks with some broader scope. And we've got some really interesting ones lined up this year. I'm really excited to tell you. Christine, who is typically the MC for this event, she was selected to do an experiment for Artemis 3, which is the LEAF experiment. So she'll be telling us about that. Joya Massa, of course, is ubiquitous. She is the plant person at KSC, and she's going to be telling us about the gaps and challenges for space crops. In between, we'll have a couple of presentations punctuated by some coffee breaks. We have a longer lunch break so everyone can take a minute to relax. Feel free to join and leave this webinar as needed. It should be running for the full 8 hours of this event. And then at the end, I'll wrap up with a few comments. Tomorrow, we're going to be doing much the same, but note that in the afternoon, the schedule is a little bit different. The featured speaker is going to be at the end. So we'll have a bunch of the shorter presentations from everyone else in between. As a note, this year we've got 30 presentations in total, 4 featured speakers and 26 regular speakers. So this is definitely the biggest version of the Space Ecology Workshop so far, and we're excited that it's expanding every year. And in fact, we did have more presentations that we weren't able to accommodate. So I'm grateful to you guys for submitting. Sorry if you didn't make it this year, but of course, you should definitely try again next year. And if you did present this year, it's always great to come back and give us updates. However, a more serious note, several of our presenters, including Joya Massa, live in Florida, and I'm sure you guys have all heard the news about Hurricane Milton. It's a bit of a mess down there, so there's a fair chance that some of our talks, the speakers won't be able to show up. In that case, I have a couple of people lined up to do substitute presentations, and we're going to try to plug them in as if we can. But if not, we'll just leave a gap in the schedule there, and we'll just resume at the next time. All of the regular speakers have 20-minute slots. So at worst, you should be waiting 20 minutes. It's an extra coffee break. Okay. And lastly, tips for the day. It's kind of the regular Zoom etiquette stuff. Well, except for that this is a big webinar. So all of the attendees, you guys have your mics and your videos off. But I definitely welcome you to chat around in the chat box. Like I said, that's going to be saved. So if you need to find something, you'll be able to do that. Keep an eye on whether you're sending your messages to a specific person or to a group like hosts and panelists or to the general audience. It's a pretty easy mistake to make. If you have questions that you want to give to the speakers, please put them in the Zoom Q&A box. And I guess I will be selecting those to read to our speakers so that they can answer. Hopefully they'll all have 5 or 10 minutes of time at the end of their talk to do some questions. But we're going to try to keep it on schedule today. So it's possible that you won't have your question answered. And in that case, I strongly suggest you follow up with the speaker. Like I said, we're not doing a mural or a breakout room. Coffee sessions are just going to be on. I'm not giving you coffee either, so you're going to have to handle that for the coffee breaks. And yeah, we're going to record the workshop and the chat log. If you've got issues, you should message me, and I'll do my best to help you. With that, I think that's everything I've got. So let's see. Christine, are you around for your presentation? I actually don't see Christine. I'm kind of not surprised that she's not here because we had some sort of last minute rearranging on on the big featured speaker slots. So, all right, here's the backup one. I have a video from Dr. Caswell, who is in Australia, and she was unable to join us because of Australia time zone differences.
Patrick,
Uh,
would you like me to go now? And Chris can maybe
actually,
go later?
yeah, that would work great. And welcome, Joya.
Okay.
I'm really glad to see you're able to join us.
Well, I have the flu, but I have power and internet. So let me get my computer back on my computer stand and I will get going. Yeah, it might be
Wow.
good because I don't know how I'll feel later. So,
Well, yeah, that would be fantastic. Thanks for helping us out with this one, Joya.
oh, my pleasure.
And really glad that you're here. It's fantastic to hear from you.
Yeah, we were very lucky. I, we didn't even lose power. So,
Yeah,
okay.
seriously, that's a miracle.
Um, okay, let me see. I think I've got my presentation. Not sure how good it's going to be because again, I have the flu, but I'm gonna try really hard not to cough. So let's just see. Okay. I'm going to share my screen. Are you able to see my screen?
Yep.
Okay, great. Um, I'm not going to share my video because I don't want to put you all through that. um, but okay, so good morning, everyone. Good afternoon, good evening, depending on where you are. And thank you, Patrick, for inviting me to speak here to you all today. It's a pleasure. This has been a wonderful workshop over several years, so I'm just delighted that it's continuing. Um, So my co-authors are Dr. Ray Wheeler, who I think we all know. He retired earlier this year, but he's got emeritus status at Kennedy, and he's also part of the LEAF payload that you'll hear about from Chris, hopefully later on today. Dr. Matthew Mickens, who's one of our project scientists at Kennedy, and Mr. Trent Smith, who's our research advisor in space crop production. So, you know, not watch because I probably did a better job there. But why are we growing plants in space? And I apologize, it says my internet connection is unstable, but I'm hoping, are the slides advancing okay?
Yeah, so far we're on why grow plants in space. So,
Perfect. Thank you.
yep.
Okay, I just want to make sure. So first of all, the food. So this bottom left image is astronaut Mike Hopkins showing a typical meal on space station. This is actually a Thanksgiving meal. So it's freeze-dried or individually packaged. A lot of the food is thermostabilized. The food quality is really good of this packaged diet. However, if you store this food for a while, as we may have to on Mars, and depending on the storage conditions, for instance, if you don't have refrigeration, the vitamins can break down, so the nutritional quality starts to degrade, and the food quality in general, the texture can also start to degrade. It's not an issue on space station because we have regular food resupplies there, but it may be an issue for a future Mars mission. And so we are looking at, supplementing this diet with fresh produce to increase the vitamins, but also the quality and the variety. Dietary variety also is a major issue in terms of having people happy with their diet. If you're eating the same thing over and over again, you start to lose interest. At the same time that we're growing plants for food, there are good indications that, at least for a lot of people, they can provide psychological benefits. And we're collecting data on this now, and I'll talk a little bit about that. And then, of course, the environmental control and life support systems benefits, which, are a little farther down the road because they're going to require more plants than we're currently growing, but will become increasingly important over mission duration. So When we think about our space crop production vision, our vision is to ensure food system security on long duration missions beyond low Earth orbit. And by food security here, we mean the condition in which the crew have continuous access to sufficient safe and nutritious food, which meets both their dietary needs and preferences in order to maintain their peak health and performance. So there's a lot to unpack there. It has to be nutritious, it has to be safe, and it has to actually taste good. It has to meet their preferences as well as just the nutritional requirements. Because if it doesn't taste good, then it's not going to be something they're going to want to eat, and have very strong opinions about their food. Um, I think this is something that we all kind of know intrinsically is that food is really important. It's very symbolic. It's very important for a lot of occasions. It's just an important thing, and so the food system has to be something that you want to eat. So in the near term, as I mentioned, we're looking at nutrient supplementation of the packaged diet. So near-term missions are going to primarily rely on physical, chemical, regenerative ecological systems, like what's done on the ISS, where a very large percentage of the water is recycled, and this packaged diet, this stowed food with some supplemental fresh food. And when I say near-term, this will be, going out to some of these early missions to planetary surfaces. But long term, we want to start to move in that direction of Earth independence. So we need to grow the calorie-rich crops. So long term, we're going to have bioregenerative life support systems, including plants, expanding over time to increase mission autonomy and Earth independence. So what this looks like in a pictorial view is where we're at here. So right now we're doing research on the ground and the International Space Station. And we're doing research on crops and crop production to supplement the food system. For Gateway and for the early lunar outposts, again, we'll be doing crop research. And then, these early lunar outposts will hopefully have some small supplemental nutrition systems, but it won't be a huge amount of infrastructure. We are going to start to test crop production on the ISS with the Ohalo 3 hardware, which is our crop production test bed currently being built for ISS. And for Mars transit mission, we believe that crop production system or something very similar will be used on the transit mission. For early Mars missions, again, we'll start to have crop production to supplement the packaged diet. But then for later lunar settlements and Mars settlements, we want to move towards the sustainable human presence. So we'll actually have more of the processing of the crops, and more integration into the life support systems, and just increase that over time. So when we think about these crop production challenges, we have some that are really specific for low Earth orbit or deep space, the lack of gravity, the behavior of fluids where water and air don't mix well, lack of convection without gravity. Some that are kind of surface specific challenges like dust and regolith. This may be an opportunity, but in the near term, it's a challenge. This is a hazardous material that we don't really want in our habitat. So maybe we need to figure out how to get some of the minerals out to use to help grow the plants, but not bring the hazards into the habitat. We're going to have partial gravity, and we don't really understand what that will do in terms of growing our crops, providing water to root zones, a lot of the challenges that we have there. Shared between these two environments, we'll have hazards such as radiation, operating at reduced pressure, hazards of micrometeorites in any environment, water recycling. Down at the bottom, we have some crop-specific challenges that are relevant for any terrestrial agricultural system. How do you have good productivity of your crops? How do you tolerate the stresses of your environment? How do you optimize for your specific environment, to make sure that the crops grow well with the constraints that you're providing them? How do you schedule your crops effectively? Crop scheduling is something that It's a really important kind of systems analysis thing when you're thinking about it, because, for instance, you don't want 30 heads of lettuce on one day and no lettuce for the rest of the month, right? But you also don't want one wheat head being produced every day, right? So depending on the crop type, you need different scheduling approaches, and you've got to work all of this in a really pretty tight space constraint. And then in the center, we have kind of our unique challenges. How do we grow very compact plants?
Mm,
So those
seems like,
are big concerns. Is it working? Okay.
yeah, it was just a momentary blip.
Okay. Yeah, I just see my connection is unstable. So we have specific food safety and microbiome concerns. We're trying to get, key nutrients out, especially to supplement the packaged diet. We want to do all of this sustainably with, some unique non-biological stresses and limited resources of our vehicles, whether it's water, air, crew time, all of our resources are limited to some extent. And then we also have crew time as a concern. How do we do all of this without taking up a tremendous amount of astronaut time, especially when we're on the surface of Mars? They need to be out exploring Mars, not doing subsistence farming. So that's where we're going to have some opportunities, like robotics and automation. And then waste is another concern, and that's where, this community's work really also plays a role, because it's not a waste, it's a resource, but how to recapture those resources. So currently on the International Space Station, we have some large plant research capabilities, the veggie and the advanced plant habitat. And these are really our systems that we are learning from. So veggie is a very simple system. It's more like an astronaut garden, low power, low mass, but a lot of crew time required. Excuse me. Advanced plant habitat, on the other hand, we call veggie's big sister. This is really a plant physiology research chamber. It has over 180 sensors and control systems for every aspect of the environment. So we're learning lessons from both of these. And, future crop production systems are not going to be as simple as veggie. We've realized there are some things that we do need to control, and there are some things that we need to automate a bit more to reduce crew time, but they won't be as complex as APH, because we don't need the spectrum of conditions that you need for research, for production. Production has a narrower range of conditions for optimal crop growth. So within veggie, we've been able to test a lot of different aspects, and one that, I haven't spoken as much on is the crew engagement. We really were able to use the veggie as a mechanism to start to assess crew um, behavioral health and the potential psychological benefit of having plants growing in this extreme, and unique environment. And you can see here, we had a lot of crew involved. They generally are smiling when they're taking photos with the plants, which is quite good from our perspective. Um, and we were able to do assessments. We also heard from the crew how much they love the aroma of growing the plants, and how that's something that they don't find in that environment otherwise. And when they said when they open the bellows of veggie or the door of advanced plant habitat, it's like being in the produce section of a grocery store or at a farmer's market. So it's something they really miss in that environment. So we've done some surveys of the crew with the veggie system and some APH experiments. And we've seen that the amount of interaction and responses varied widely by individual. And this isn't surprising. I mean, you can take any population of people that you know, and some of them love plants and want to be in nature, and some of them really could care less. And if they never leave their basement and only eat Hot Pockets, they're fine. It's just like any other group of people, and the astronauts, I think, are the same. But we did see overall that enjoyable tasks had a higher impact than non-enjoyable tasks. And that the crew, when they were interacting with Veggie, they viewed it as engaging and meaningful work. They all really understood the importance of having plants in an off-Earth environment. If they found that it had a positive impact on their mood, their well-being, and their relationship with crewmates, and it enhanced their subjective connection to Earth and their desire to work with veggie through harvest and consumption. And even the people that didn't really want to spend a lot of time with the plants were happy to eat the plants when they were available. We also saw that veggie was a positive source of sensory stimulation, and you can see that graph on the right here. Um, with very high impacts on sight, smell, and taste, and a little bit lower on touch because we actually have the crew wearing gloves when handling the plants, and I don't think they like that very much, but that goes back to the food safety concerns. When we think about the food safety concerns, we can do studies kind of like this one. So this was a study about red romaine lettuce, and we compared our lettuce grown in Veg 3A and Veg 3D to lettuce grown in the supermarket, the market lettuce there, all the way on the left side of these plots, and our lettuce grown in controlled environment chambers at Kennedy. And you can see that our Veg 3A, which was cut and come again, that's what the CACA stands for, repetitive harvesting of the same plants, um, showed pretty similar and even lower results than what we saw from the supermarket and the growth chamber in terms of aerobic plate counts and also yeasts and molds, which is the green graph. So this is bacteria in the blue, fungi in the green. But Veg 3D, which was one of the next grow outs having that type of lettuce, had higher levels. And so, this wasn't due to any specific factors that we could identify. It just maybe happened to be what was going on the space station at a given time. So it's very challenging to control food safety in that environment. We're not saying that these high aerobic plate counts or the high yeasts and molds are unsafe, because that's not true. But this is an indication that there is more microbial activity in these levels. And so, one thing that NASA's trying to kind of wrap our arms around are what what is food safety specifically for produce. We look for specific pathogens, and we actually haven't identified too many of those associated with plants, and certainly not at levels of concern. But are aerobic plate counts something good to assess, or are these not really relevant? So, um, you know, living food, plants, has higher microbial levels generally than non-living types of food. And so this is something that, we are actively working on, trying to really understand how to control food safety with crop growth. And it's not a simple challenge. We also can study the microbiome, and this is an image of the Veg 05 tomato microbiome. Now, Veg 05 had a lot of challenges. We had uh, poor fruit production and a lot of loss of fruit due to water stress, and we had massive adventitious root formation in these plants. And the adventitious roots ended up being kind of microbial repositories. It was a very interesting but stressful experiment for us. What we can see here with this sort of microbiome heat map is that there are some differences between flight and ground, but there are larger differences between tissue type. I think my internet's going in and out. Apologies. Um, there are larger differences between tissue type, and we can see very high microbial levels in the roots, which are the parts of the plant exposed primarily to water, and the adventitious roots, which also, especially in flight, had a lot of water. Um, So we had higher community diversity in those than in the, the purely above ground fruit and leaf samples that were sort of saturated with water. We've had some pathogen issues. So this was from our zinnia experiment, which was our Vega 1C experiment. And we had these sort of very abnormal stressed plants. You could see in the left, we had leaf curling, the epinasty, and we had the little guttation droplets along the edges of the leaves. And in the right, we had some very, very small stunted plants in the back, and these in the front did not look happy or healthy, and they eventually got fungus, which you can see here. And this ended up being due to the fans shutting off in veggie. So we know that forced convection is required for microgravity, and we didn't realize that the fans were actually off during a period here, a pretty extensive period. Water built up, and it led to opportunistic plant pathogens colonizing these plants, which was pretty pretty awful. Um, However, when the fans were restarted in this experiment, and plants were cleaned, some of the plants survived and grew very well after this point. So watering and water and air mixing is just a real challenge, and keeping track of, whether or not you have good convection is really important in this microgravity environment because these pathogens are up there. We have no idea where this Fusarium oxysporum came from. We tried to do an extensive, tracing of this, and we never were really able to find out. But there are a number of plant pathogens on the space station. Sometimes they can come from within seeds. We clean the outside of seeds when we send them, but we don't, have a way to clean those endophytic organisms within the seeds. Everything else we send is as sterile as we can make it, but you know, we're not sterilizing the crew. We're not sterilizing everything. A lot of the hardware is set up in clean rooms, but they're not perfectly sterile. So there are pathogens up there, and I think this is something that we definitely need to consider as we're designing future life support systems. We can't get rid of everything, so how do we keep things in the right balance and under control? So for APH, advanced plant habitat, we've had some crop experiments. This is the radishes by Professor Carl Hassenstein from University of Louisiana Lafayette, the PHO2 experiment, and the astronauts got to eat some of these. They also got to eat the Espanola improved hatch chili peppers in PHO4 experiment, where they were able to answer the New Mexico state question of did they like their chilies red or green? And I think they liked them both ways. And coming up next, we have our PHO7 experiment. So this is led from our team at Kennedy, and we are launching this hopefully at the end of this month. We're pretty excited about this because in this case, we're actually going to intentionally look at water stress instead of unintentionally looking at water stress, which is what we've done with a lot of our previous experiments. So um, we're going to grow the outrageous red romaine lettuce and intentionally apply a flood treatment, a constant drought, and then an intermittent, water and wilt treatment, and compare the control grown plants. We're going to have barriers to separate them. That's what you can see kind of dividing the shoots here from the overhead view, to keep the leaves from touching. And we have separate quadrants in APH, so the root zones are isolated. We're also going to be able to extract roots from this experiment. And we want to look not just at the plant responses, but more importantly at the microbiome responses, because we want to understand what's the best failure mode for the ecosystem as a whole. We need to figure out when we're designing these systems, should something fail with too much water? Should it fail with not enough water? What are the short-term and long-term ecosystem impacts of those types of failure modes? So we're really excited about this. This will be a series of 3 28-day replicate growth experiments so that we can get enough sample sizes to really understand those impacts. So our key crop plant takeaways are that our data indicate the astronauts enjoy growing and eating plants in spaceflight, and our crop yield is similar between the ground and flight when the plants are not stressed, but that's a challenge. We do see some differences between ground and flight plant nutrient content, but they're not consistent between species or experiments, and they're influenced by things like crop type, harvest scenario, and other environmental conditions. Our microbial counts and compositions vary between flight and ground grown plants, and we see these microbes typical of the space station environment, often associated with ISS grown plants. We've only seen one human pathogen, which was a Staph aureus, on one plant and one experiment, and it was only below levels of concern. But we have to do our due diligence to try to understand if those human pathogens are there and how to keep them out. Because, it's not easy to wash your hands in space after you go to the restroom. So it becomes a challenge in this environment. We see that the microbial communities vary between shoot and root tissues, but not as much between flight and ground. And overall, I would say that our plants adapt well to the space environment, and to date, the space-grown crops are safe to eat, nutritious, and highly acceptable to astronauts. So we've developed a series of gaps, and this is a living list, and we're actually in the process of kind of reviewing and updating these right now. But we have about 65 different knowledge and technology gaps in 13 areas related to crops, ecosystems, and the hardware, with our highest priority gaps in these areas that you can kind of see listed here. And then this is just sort of a screenshot of a few of our gaps lists. Lack of ability to effectively provide adequate and uniform delivery of water and nutrients to root zones, for instance. That's one of our higher priority ones. So if we have some knowledge gaps, some examples that are relevant for bioregenerative life support systems, we need water and nutrient delivery approaches at different gravity levels, and so what works for the space station may not work for the moon and may not work for Mars. So we need to really understand those better. We need non-invasive plant health monitoring and pathogen detection technologies. We have to kind of understand how plants are reacting to the environment and if there is stress before it becomes limiting to growth, and be able to respond and mitigate those stresses. For instance, a water stress or a nutrient stress or a pathogen stress before it starts to really impact growth. So we're doing a lot of work with visual sensors, but types of chemical sniffers, I think, are also going to be important to make this integrated picture of what's going on with the plant. And then we need effective options to recover resources from inedible plant wastes and human waste streams. And we not only have to recover those, but then we have to dose them back to the plants at controlled amounts. So how do we, get out the nitrate or the ammonia, and then how do we add that back into the plants in known dosages? It's going to be a really important thing. For space crop production, some knowledge gaps are, specifically that our library of crops tested is very limited. We've only tested a relatively small amount of crops in space, and about a third of those or more are brassica families. So we need more diversity of different crops and crop types, and we want diversity for robustness and resilience of our system. Because if you have a disease that impacts only brassicas, for instance, that could take out a huge percentage of your crop library. In terms of the plant microbiome and food safety and plant health, we're probably going to be growing crops hydroponically in surface habitats, at least near to midterm, and potentially in microgravity as well. And we have many questions still around hydroponic water and nutrient delivery, and very few data on the hydroponic microbiome. So that's an area that I think we need a lot more work in. And then exploration atmospheres, the reduced pressure that we're going to be growing on the surface of the moon and on the surface of Mars, so that astronauts can easily EVA without doing a long pre-breathe at the lower pressure. It's largely unstudied for plants, and data from the University of Guelph indicate that plants will grow okay in these environments if you keep the partial pressures of CO2 and O2 high enough. But we really don't have data on plant microbial interactions, responses to pathogens at these reduced pressures. So a really big area of work that needs to be done. And unfortunately, not very many people have the facilities to do these types of experiments. So that's limiting in this area. And then the similar case is true for the deep space radiation impacts on seeds and plants. And while some of our team members and team members around the world have done studies of different radiation sources, um, most of these are at a rate that isn't really the same that you would see. So maybe the total dosage is similar to what you would see on, say, a mission, a 6 month, journey to Mars, but the rate is over 3 hours instead of 6 months. So until we're actually in this deep space environment on a platform like the Gateway, we may not have a good way to mimic this space radiation. So one area that I think there is some opportunity is actually selection of space plants. And it could fall, be targeted breeding or engineering, falling into different categories. And we've identified a list of these categories, and here are just a few examples, but plant growth and development, getting compact plants with high harvest index, uniform growth. Plant physiology is an important area. We want stress tolerance, especially to some of our unique stresses. And for instance, if we are going to be recycling urine, then high sodium tolerance might be a really great characteristic for our crops to have. For nutrition, we want high normal levels of the things that we want, the nutrients, the beneficial phytonutrients and antioxidants. And we don't want any anti-nutrients. For organoleptic acceptability or sensory acceptability, we want good flavor, good texture, intense flavors, appearance, color, aroma. We don't want like displeasing aromas, right? Because if they're in the space habitat and you just hate the smell of that plant and that's all you're going to smell all the time, it could be really bad. And for post-harvest, I think there's some really interesting opportunities there, having a good shelf life, whether on the plant or off the plant, reduced processing, when you get to things like grains, for instance, having them much easier to thresh out of the seed head would be great. Easy degradation of inedible biomass. You're not withstanding gravity, 1G anymore, right? And so much of the cell wall structure of plants is designed to stand up to 1G. So could we reduce that cell wall structure, for instance, to make that inedible biomass less recalcitrant, easier to break down, and maybe channel those, proteins or products or whatever into more food or more beneficial compounds. Um, if you don't need to withstand 1G, then you need a lot less structure, perhaps. So I think there's some real interesting opportunities in post-harvest. So our space crop production team has developed this roadmap, and it's an eye chart, so I don't want you to read it all, but these are the different hardware, technology, crop science, ecosystem, and human factor tasks that we need to be studying or things that we are actually doing in these environments. Um, we have some on the ground, some on the space station with Veggie and APH and the upcoming Ohalo 3 hardware. And then, certain tasks that will occur on Gateway and on the lunar surface. And again, this is all a plan. So, we hope we will be able to do a lot of this, but much of this remains unfunded. Um, we have different funding sources that we're kind of quilting together from different groups, and then we have partnerships, and that's really what's very important. Our USDA, Agriculture Research Center partnerships, partnerships with the Australian Centre of Plants for Space, which you'll hear a little bit about during this. Canadian Space Agency, German Space Agency, Growing Beyond Earth, which is our education and outreach program with middle schools and high schools around the world. We have some Space Act agreements with various universities, as well as some work with the DoD. So we have to do all of this as partners because nobody can do all of these things alone. So I will stop there, and I'd be glad to take any questions and just thank the teams and the funding agencies for this. So I will stop sharing.
Thank you so much, Joya. That was fantastic. I really appreciate you powering through the illness. Um, yeah, we have a bunch of questions actually, and I have some as well. We've got
Okay,
10 minutes to do it. So
great.
let's see. One of the first ones is from one of our co-panelists, and Kevin wants to know, I'm interested in applying my research on plant microbe interactions to growing plants and regolith, specifically how to use microbes to extract regolith nutrients. Is
Mm.
NASA currently interested in this? And if so, do you have any advice on applying for grants to fund this sort of research?
Well, I mean, we're definitely interested. I personally think that extracting the nutrients from the regolith and then reapplying them to the plants is going to be a lot safer than trying to grow the plants directly in the regolith. Just because NASA's spending, millions of dollars to try to keep regolith out of habitats. So, the idea of growing in regolith directly, there's a lot of challenges for that. Um, in terms of funding, you just don't know. There should be a space biology call out sometime this fall. Whether something like that topic will be a part of it, I'm not sure. I'm not really privy to any of those conversations. Um, so yeah, I think it's just kind of keeping your eyes open. You can sign up at inspires.nasa.gov to get notifications of relevant calls. There's a group called TRISH, the Translational Research Institute for Space Health, and for things directly related to human health, they do fund some kind of out of the box thinking. There's also the NIAC call, NASA Institute for Advanced Concepts, which funds, pretty far out there ideas. But even smaller projects via state space grant consortiums. Every state has a space grant consortium, which funds student, research and student engagement activities. And so you can do an unsolicited proposal to your state space grant consortium if you're in the United States, and get a small amount of funding potentially to fund your research, or get funding to travel to a conference and present your research, for instance. So definitely look at whose state space grant consortium you have, and talk to those folks and just see if there might be opportunities there. Um, and then, if you're not in the US, there may be different funding sources from other space agencies, but you can almost always write an unsolicited proposal. It's kind of a message in a bottle, right? It's a bit of a wish thing, but you can always send those in and maybe get some feedback on it. And, so I encourage people to start writing some proposals for their ideas. It's a great skill, and it can only get better with practice.
Yeah, thanks for that, Joya.
Mhm.
Funding is always the top of people's mind, and those are
Of
some good
course.
suggestions.
Yeah.
Um, Shazari and I both have a question about Ohalo. I think this is the first time I'm hearing about it, and I'm curious if you could tell us a little bit more about that and how it's different from the existing facilities. And also, Cesare wants to know when the first flight is planned for Ohalo.
Sure. Yeah. Great to hear from you, Cesare. That's awesome to have you here. Um, at this point, I believe Ohala will be flying in 2026 in the summer or fall. So Ohalo is really a space crop production test bed. So it's going to be on the ISS, and it's a quad locker, so kind of similar in overall size as APH. But as I mentioned, it's really a crop production test bed. So it doesn't have all of the control systems of APH. So it actually has a larger growing area. APH growing area is only 0.2 meters squared, and I think Ohalo will be almost double that. It won't have as much height as APH or Veggie. Um, we are going to design, well, we're designing kind of the base hardware, and our team at Kennedy's been working on this for a while now. It has, lights and fans, and it's interesting in that, so APH is kind of a closed system. It recycles the atmosphere inside, and it has to actually add CO2. So we have to fly CO2 for APH, whereas Veggie is an open system, and all of the environmental control is from the space station environment. Ohalo is kind of doing a hybrid approach to this, where it will be recycling the water inside, but taking up CO2 from the cabin. So this will be the first time we're testing that, and we're real excited about that. And then in terms of the water and nutrient delivery, we have a number of teams that have designed what we call hosted hardware. And so two of these are in the latter phases of this kind of, I guess it was a grant competition for this hosted hardware. And so we have two different groups, Utah State University and Sierra Space, that are designing kind of root zones and volume optimization for inside this Ohalo. So they have some different approaches, and hopefully we'll get to fly both of those. And we're really excited about looking at these different options. But I think that's about all I can say at this point. But yeah, be on the lookout for it.
Okay, that was pretty interesting. Um, Ben Sykes wants to know, have there ever been experiments on the ISS where you planted multiple species of plants in the same box simultaneously?
Yeah, we've done that a few times in veggie with mixed cropping. We did this once in Veggie 3D and E, where we actually had mixed cropping in both veggies with a slightly staggered start. So we had Mizuna mustard and then red and green lettuce. And this gave the astronauts a chance to look at what continuous harvesting would look like, because we were doing this cut and come again harvesting. So we planted them about 3 days apart, and then they could have some salad crops every 3 or 4 days. We've also done multiple, different species. I think we've had up to 5 different types of plants in veggie, which only hold about 6 plants. So you get some competition, of course, for light and other resources. So if the growth rates are very different, then you can get some shading. But other than that, it's been fairly successful. Um, Also, European Space Agency has done some tests with lentil, mustard, and radish seed germination all at the same time. And of course, the Russians have grown different crops in the Lada system at the same time. And we've also done this in APH, where we had both Arabidopsis and wheat planted at the same time for the validation test. So I think there's been a fair amount of multiple cropping. But I don't know that anyone's really looked at, things like the plant-to-plant interactions for those experiments, which I think would be a really interesting area to study more.
Wow, I'm surprised to find how many multi-plants experiments there have been. Got several more questions in only a couple minutes. I'm gonna combine a couple. Were there any significant findings on specific species of microbes that became perhaps more pathogenic compared to the same microbe on Earth? And do you think that plant resistance to microbial and viral pathogens is one of the more important traits?
Yeah, that's a great question. I do think that that's going to be really important. And I just think we don't really understand it very well yet. So, we haven't seen that many pathogens, but the Fusarium oxysporum that we did see, we tried to replicate on the ground what we saw in space, and we really weren't able to get as severe an infection as we saw in space with that lack of convection and the building up of water, even with the same microbe, from the plants that were brought back. When we treated zinnias on the ground, and Professor Andy Sugar at University of Florida did these wonderful Cook's postulates plant pathology experiments, and he put these plants inside a bag with 100% humidity, and even then we didn't get as severe infection. So we have a couple of we have some recent investigations with plant defense responses. Dr. Anjali Iyer-Pascuzzi at Purdue just flew the PH06 experiment looking at plant defense and immune responses in young tomatoes and APH. And so she's analyzing those data. And then Dr. Andy Sugar has an upcoming investigation with a powdery mildew on Arabidopsis. So I think we'll be getting more information on these things now that, like, the real experts are doing this type of research. But I do think that that is really an area that we need to spend a lot of attention.
Okay. That's surprising that that it was in fact much worse on the International Space Station.
Mhm.
Well, that's all the time we have for questions. Trying to stay on track. Joya, seriously, thanks so much. It was a delight to hear from you. Really glad you could make it, even though you're sick and in a hurricane. Thanks again.
Thank you.
Um, yes. So next talk, we've got Yvette Gonzalez, and she's going to be telling us about bioastronautics research in suborbital space flight, parabolic, and cave analog environments, which informs sustainability. Hey, Yvette, take it
Hi.
away.
Hi. I'm going to share my screen. I hope it works. Let me open up. If this is gonna, it should automatically, right? Let me choose my screen.
I think so. Yeah.
Hmm. Let me try it again. For all the work that I'm about to share with you, technology is not my strong point. Aha. Okay. Here it goes. All right. There it is.
Yeah, we can
I
see that.
want Yeah, okay, great. I wanted to thank you for hosting this conversation and definitely for being able to have this discourse and bring these colleagues together. And thank you so much, Patrick, and your team for everything you've done to prepare us. I am going to just start quickly. I'm Yvette Gonzalez. I'm currently in Germany. I'm traveling quite a bit, and then I'll be stateside soon. So I'll be catching up with a few familiar faces I see here today. So thank you so much for the conversation. I'm sharing today all the bioastronautics work I've done the last 10 years in orbital, suborbital spaceflight, parabolic flight, and a caving analog, because I want to share how that informs our ability to sustain life off Earth and why we're doing that. But I'm going to start because I am also from Texas, Arizona, and I am a descendant of Aztec and Mexica ancestors. So I do a grateful acknowledgement to acknowledge the indigenous peoples on whose ancestral homelands we gather or we're currently located, as well as the diverse and vibrant indigenous stewards of land, skies, stars, and water. So we honor you for joining us today and for joining us even for the recording. The goal today is really that I want to share not just what I've been able to do with my teams, but I want everyone in this audience and who watches this in the future to know they can have access to these environments and what each platform I'm about to share, actually how it informs the way we do research, how it changes how we do research and what our methods are, and how we consider that in the future. Also, why does it matter? Why are we doing this? How are any of our research questions relevant? And how does it all push us to rethink how we share all these limited resources? Not just what we're going to be sharing off Earth, but what do we do here on this planet? And hopefully that will inform or give you some information and knowledge on how that might help develop programs, policies, and certainly ethical considerations about how we build these ecosystems together. So I start with the bioastronautics work that I do. It's basically for me bringing the nexus of biological, behavioral, engineering, and space research together. So the bioastronautics that I work on uh, takes the human factor, yes, but we are still conducting both human-tended experiments and studies and asking all the large questions. So Goya's presentation was quite accurately encompassing everything that my teams and I do. We look at all these factors, and then we all come with our own discipline and we share our perspectives, and then we learn together what might work off Earth. So here I'm just showing you that as I started, I joined a Bioastronautics Institute so that I could actually start to learn outside of my discipline of epidemiology and public health. What does bioastronautics mean for people planning to go off Earth? What does it mean to survive within your own spacesuit? What does it mean to do survival training with a team? And then what does it mean to experience microgravity in a gravity offset harness for lunar, Martian, and even potentially working on the outside of the ISS? That said, one critical thing that most of us get a privilege to do right with our teams is to learn from them their disciplines and their background. So through this institute, it was also important, and they offer this kind of courses today to look at your discipline, but then learn planetary geology. What is it that we are going to be exploring on other planets? What kind of soil will we encounter? How do we investigate that? And how does that inform other science? And it was important to get those skills because I didn't realize that building on that, my human health experiments, my epidemiological investigations were also necessary to integrate into the thinking, the wider thinking of astrobiology, the thinking of geology, and why does that all interconnect, and what's the reciprocity between all of those disciplines. In fact, two actually of the peers from that training had hosted the first space research caving analog, where 7 of us conducted a lunar space research analog last year, lived in this beautiful cave for 7 days. And as the head of the research, it was 21 experiments from all different disciplines. And to integrate that into 7 days was looking at a human health suite of studies: geological, geoscience, geophysical, geotechnical, astrobiological, and on top of that, all the considerations that Goya, her talk triggered all of these kind of preparations, which is food allotments, planning for necessary nutrition, energy levels, maintaining and monitoring that within the system, looking at water resources, looking at our sanitation. So you're right, we don't look at it as waste. We look at that full nexus of food, what do we call it, farm to feces, where what is it that closes that loop and how can we be efficient? And so we took a lot of this research and combined the efforts together, which was really fascinating. And one of the ones I want to highlight here is for the microbiome, the human microbiome. We were doing jaw movement. And I was working with Dr. Mona Nasser. She is the PI from the University of Plymouth. And you'll see here on the left, I set up in the base camp to conduct these so that I had a proper amount of light and recording. And what we're doing is she's demonstrating where the dots go on the face. And every morning after breakfast, I had all of the participants chewing gum and recording to see the markers on their face of stress levels that were either increasing or decreasing along the analog. And on the right, you'll see that this is actually our co-PI Prashanti created a kind of diagram for me to work through so that I could understand exactly where the placement was, because we were trying to inform AI models that might be able to offer us technologies that we can use in space and that we can use off planet. Simultaneously, we have all these Earth analogs, laboratories, and facilities that we're using on Earth. We do have the ultimate laboratory, which is space. And getting access to that is something that has been really a privilege the last few years. So I start with the Virgin Galactic suborbital flight. We had that last year in November, and I was in charge of the human health monitoring through a biomonitor. And the interesting thing is, some of you may have seen that CSA had this on station on the ISS with astronauts testing it up there. We tested it before it went on station to get the test readiness level advanced. But then here, what was exciting is it's the first time that the Astroskin was used on takeoff, re-entry, and landing. So monitoring human health in that capacity has been fascinating, and we're currently analyzing and getting ready to publish papers on that. But here I show you, even for the few minutes, it was a 90-minute flight, wearing the Astroskin underneath a flight suit. It's a passive, very non-invasive way to monitor our humans' health in space, especially when you have other monitoring devices on. She had several, and conducting other experiments external to the body. So it was quite really an investigation of and a study of the human interaction and what can we accomplish passively while we're distracted with other procedural operations. The next one was the Axiom Mission 2 last year. And we worked with a Saudi Arabian research team called Nebula R&D, which brought together dozens of international experts. And our team analyzed and put together 8 chapters in a book that has just been published because we had a suite of neuroscience technology interventions and equipment. And it's quite interesting to see that when you use multimodal devices, what kind of information, a holistic picture you can get of an experience in microgravity or different hypergravity. So I will share the link after for that if you're interested. And then Polaris Dawn. We just had the return of Polaris Dawn, the first standing EVA commercial flight. And the standing is that there was a partial EVA, which meant that not only were all the equipment and experiments on board exposed to the vacuum of space, so were the people. And it would be fascinating to share in the future what the impact was for ours. My team was with the International Institute for Astronautical Science and Nebula RD, and we were doing the pupillometry, so looking at changes in the eye. So we're really excited to see the results of what happened pre and post SEVA. So all this, we talk about sustainability, you know, and with the term is used and overused, but really it's what is the ability for us collectively to sustain life, biologic or human, off Earth? What are those abilities? What do they mean? And what are we doing with this science to get there? And I know I'm rushing through all this because there's so much, but I really wanted to address these. The access to these environments. It is not impossible for early scientists, mid-year scientists, and late career to be in on new questions in research, asking and evolving innovative questions. And these environments that I've showcased here, even the parabolic flights, suborbital and orbital, if you would like access to those, I'm more than happy to show you how it's done. And I'm going to share some links in the chat. But the first thing that we did as a team, we went to all the launchers, got on all the lists so that we could get notified of calls for proposals so that we know when there's going to be an opportunity to send something into space. And when those calls come, the one thing you should be doing behind the scenes is getting your work ready. What's your research question? Why does it need to be in a microgravity environment, and what does it contribute to science, right? You always have to think about what justifies this floating, and what does that mean for the advancement of knowledge for you, for your team, but also for humanity. We also have to consider, what does it take to be on each of these platforms? You know, I have to say that when we look at all the environments where we're testing, we're collecting information, right? I'm collecting data here, some samples here. What am I going to do with it? I'm analyzing it with my team. I have so many teams. I never work alone. To Goya's point and to Patrick's earlier point, we aren't working by ourselves. It takes teams, it takes entities, it takes academic institutions, it takes research institutions. We're constantly getting different perspectives, getting different ideas for innovation, supporting each other, bringing different cultural aspects to our research answers, and then considering more. And then we're testing again. I mean, I've taken these samples and then we go into microgravity, we do another experiment, and then once again we reiterate. But more than just the low-hanging fruit of publishing, we really are trying to see, what is the applied case? Where do we put this information? And the greater good, right? The why these studies are relevant, we really hope that someone will grab that information and it will be useful for advancement of their own technology, maybe answering questions for a Mayo Clinic research, or for their applied use with patients to say, oh, we hadn't considered that, or we didn't know that was possible in microgravity. Now we understand something new. Maybe it will improve medical equipment and the uses of that to not waste as much. There's so many other applied ways that we hope the outcomes of all this research can be used, but most often it's, can all of this answer questions for a closed loop system off Earth, out of this atmosphere, off our ideal planet? And what does that mean for us? Yes, there's comfort. Yes, there's the what are the things we need to feel comfortable, but what are the things we really need to survive? And how does all of this meet that for us? And then we rethink our resources. in all of these settings, we're also given just what we need for that moment. We were in the cave, we got what we got, and that was it. You had to work with what you had. We went out to Flagstaff to the Apollo training site. I mean, you go out there, it's more than 100 degrees. You're working with what you brought with you. There's no going back to get what you need in town. That's at the end of the day. So you really have to plan out your days, your flight schedule, your how you're going to help your companions and your teammates. What equipment can you carry? Are you all hydrated? Where are we going to use the bathroom? All those concerns apply. And then the outcomes we also hope, when you consider all of the information that we're learning it in real time, we're sharing it in real time. And so we're hoping that that learning experience is informing somebody that could reform maybe a health policy, reform a climate adaptation strategy for Earth. But then we're hoping as well that this larger community, when we're thinking about the ecological frameworks and the ecosystems in space, that we're answering each other's questions, or we're informing them, or we're helping to advance them. And so I would say, Patrick said this, but I believe it truly, the collaboration is the most important part. No one's going to space by themselves. No one's building habitat on the lunar surface by themselves. So the more that we can share, the more that our human lived experience, and here I show you this because I want you to see visually the way I see my research. I've been a humanitarian for over 2 decades. I take from my human experience, I take from my community experience, I take from my agriculture experience, I take from response to emergencies, interacting with different communities around the earth. And then I take the space technology and the space research. And I always have to come back to that question of why does this matter and how will this help in the future? So I hope that, we can consider all the work that we're doing in that framework. And I'll stop there because I would love to have a conversation and answer any questions. Thank you.
Okay. Thank you, Yvette. That was really fascinating. Always great to have the human perspective in this because we often get sidetracked by plants. So we have a couple of questions. One of them should be pretty quick. Miguel wants to know specifically about the neuroscience experiments you were talking about. Is the data from the EEG open source?
Oh, that is actually a really good question. I think it will be soon. Part of what we did was the neuroscience suite. So right now I know because the publication is behind a paywall, and I don't know how long that will be for. There was another team that was working simultaneously, and you probably are familiar with Chris Mason and the SOMA, the Space Omics and Medical Atlas. Are you all familiar? I'll put that in the chat. And that information is open source and it's on the Medical Atlas, which is already online. So we're hoping, yes, thank you, Miguel. We're hoping that eventually all of these space flights, in fact, the Virgin Galactic one, the biomonitor, and the glucose monitoring and a few others will be added to the NASA Gene Lab and SOMA as well.
That's great to hear. Christina has a pretty similar question about the Astroskin data. Is that available anywhere? Same place?
Yeah, we're going to be adding that to the SOMA. So, and to NASA Gene Lab. So hopefully that will be open on the what is it, the open repository? I'm not
Open
really
Science
exact.
Data Repository,
Yes. Yeah.
OSDR.
Yeah.
Yeah.
So we're working towards that, Christina.
Fantastic.
Oh, also,
I have, go
sorry.
ahead.
I'll mention to Christina as well. The Astroskin is on the Caritech website, the Hexoskin, the company that makes it in Canada. They have about 200 publications as well. So just, those are open source and those are free.
Okay, that's good to know. All right, we've just got 1 minute left, and I've got a quick, silly question for you. Living in a cave for a week sounds pretty crazy. Tell me your favorite and least favorite part of that experience.
So I'm gonna just embarrass myself. So before we went in, they asked me part, and I said I'm afraid of the dark. So why the heck did you choose me? And I don't know where that fear came from, but I'm gonna tell you that that was the absolute last thing that was on my mind, and I embraced the dark. I think that my favorite part was being alone in parts of the cave. You couldn't always be alone, but in distance from somebody to give you privacy, and turning off all the lights, and in that kind of being wrapped by the earth, it felt was something very special and unique.
Wow. Yeah,
But
that does sound like a very. Go ahead. I'm sorry.
no, sorry. I think I had a delay. I'm so sorry. Um, but I also like the food. So back to Goya. Goya left me hungry after her talk, but the food was amazing. So.
Yeah, got to keep your morale up when you're under when you're in a dark cave. All right. Thanks so much, Yvette, for that. It was a great talk. We're gonna move on next to Dr. Celine Klotz. Hey, Celine, welcome.
Hi, Patrick. Can you hear me?
Yeah, we can hear you just fine.
Okay, great. Second,
I'm sorry, I forgot to introduce your talk. Dr. Celine Klotz is going to be telling us about microbial resilience and adaptations for space exploration, a step towards sustainable bioastronautics. So, looking forward to this one.
Thank you, Patrick. Yes, so today I'm going to present our studies on microbiome in extreme environments and how it can be used to develop agriculture in space. So there's a lot of studies done on the impact of space travel on the body, and in particular its evolution. So there's a lot of studies that have shown there is an impact on bone and muscle, but it also shows that the body is able to evolve, to adapt to this extreme environment. For example, it has been shown that there is that the space travel trigger neuroplasticity. It also the body becomes also more efficient in the use of oxygen and energy. And there's also studies that have been shown that similarly the microbiome evolve during space travel, and in particular there's a epigenetic change that will translate into modulation of the gene expression, which will for example, after a space travel, you can see the bacteria have increased growth, also increase DNA repair, increase resistance to antibiotics. And this is important because the microbiota has a huge effect on the body and its health. It can impact not only the digestion, but it can impact the brain. That's why the intestine is called the second brain. It can impact your intake of nutrients, your skin, how you take weight. So the impact of space travel on microbiota is also very important for human. But it's also important on the plants, because similarly to humans, plants have a microbiome that can impact their health. The ecosystem of bacteria in the soil and directly on the plant can impact, for example, the intake of nutrients in the plant. It can also impact the resistance to disease and to pests. Also impact the resistance to stress, environmental stress. For example, the lack of water, also the salinity, also the presence, for example, of heavy metal in the soil, and it can also impact the content of bioactive in the plant and in the fruit. So how can we use this microbiome to improve the agriculture in space? So for this, we developed we studied microbiome and plants in extreme environments, like for example deep sea, in the mountain, or also in the desert, where there's a lack of water and extreme heat. And we have seen that in this extreme environment, both the microbiome in both human and plant evolve, adapt, and evolve. And this leads to an increased resilience of both human and plants to this extreme condition. For example, in plants, the microbiome will protect the plant from infection and improve the intake of nutrients. And also what we have seen is that the microbiome is going to affect the contents in bioactive in the plant. So for example, we have made studies on olive trees. And what we have seen is that in millenarian trees that were in extreme environment, in the desert, we have studied the antioxidant effect of extract from olive leaf from millenarian trees and from young trees, and the millenarian olive trees had a higher antioxidant effect than the leaf from the young olive trees. And what we discovered is that because the microbiome from the millenarian olive trees produce small molecules, metabolites, that have antioxidant effects. And so because of exposure to extreme environment, the microbiome of this millenarian olive trees, they have evolved for thousands of years, and they have become particularly super bacteria. And when you transplant this bacteria from the olive trees to other plants, for example, pomegranate, the pomegranate becomes more resilient and also produce bioactives that have a higher effect, for example, on anti-inflammation. And so from this discovery, we have developed a platform of bioprospection where we isolate and select bacteria from soil and plants, and we study them. We study which metabolites they are producing, and we develop a technology to harvest this bacteria and metabolites to produce a solution for agriculture and health. For example, we have isolated bacteria isolated from palm tree growing in the desert, and we have discovered that this palm tree produce metabolites that activate DNA repair and stabilize collagen when exposed to UV. And so using this bacteria that we have prospected, we developed a fermentation technology that produces bacteria and metabolites. And what is unique with this fermentation is that we use cold culture technology, meaning that on the contrary of conventional fermentation, which use one bacteria, we use several bacteria that that collaborate, that I would say interact with each other through chrome sensing. And through this co-culture, we produce new metabolites that have higher activity. And this fermentation, through this fermentation, we can produce up to 500 metabolites. And so using these bacteria and metabolites, we can produce solution for not only for agriculture, but also cosmetics and food. So now I'm going to present you the result of some tests we have done for agriculture. So using our technology and the bacteria that we have isolated, we can produce solution not only for farming, but also for chicken, cows, horses, and also for fish. So here we used bacteria isolated from millenarian olive trees, and we tested on the rice. So in the picture you can see it's a gel. The form is a gel which contains a complex of bacteria. And we applied this bacteria on a rice field before and during rice seeding, and we compare it to other fertilizer. And at harvest, we measured the plant fertility and the yield, and this test showed that aquabiota had the highest effect in terms of fertility, and also in terms of rice grain weight, and also number of grains, so the yield, and it also had the highest, the rice grain cultivated with aquabiota also had an increased nutrient content. So this is another test we have done in hydroponic cultivation setting. So it will be a setting that would be similar to what you could find in space. So in this case, we have done the test on lettuce. So we have divided the lettuce into two groups. Control group where the lettuce was grown in nutrient medium, and the second group, the active group, where the lettuce was grown in the nutrient medium added with 2% aquabiota, which was added weekly for 4 weeks. And after the 4 weeks, we have noticed that there was a 21% decrease of harvest time, also increased shelf life, 12% increased in yield, and also increased of the quality of the salad, with increase of nutrient and some bioactive like quercetin and luteolin. So this shows that by using aquabiota and bacteria that come from that have been isolated from extreme environment, we were able to increase the yield of agriculture and improve also the quality of the food. And so this could be advantage in space travel, where the space is limited. Also, it's so it will increase the harvest and yield. And so our next project is to send this technology into space to test directly in a space environment the effect on plants. Thank you.
All right, thank you so much, Celine. Great presentation. Yeah, it's nice to hear about the microbiomes. If you guys have some questions, put them in the Q&A box. We've got 5 minutes to go through them. Celine, I've got a question for you.
Yes.
It's interesting how the microbiomes and the kinds of bioactive compounds that you can find vary from place to place. When you're choosing an environment to sample to prospect these microbes, what are the factors that you consider?
Low water, salinity, heat, and extreme and higher UV exposition. For example, mountain. This is some condition that you would find actually in space, like compared to radiation, also low oxygen, limited water source.
Okay, I think I am having internet issues right now, but if you can still hear me,
Yes.
I've got another question.
Yes, there's a question from Yvonne. So no, we haven't done on experiment with fungi, only bacteria at the moment.
Okay. Another question from Kevin is using the microbial metabolites a more effective approach than directly inoculating the plants with the microbes responsible for the metabolites?
Usually what we do, we use both. Actually, we use a mix of metabolites and microbe. What is good with the microbe is that it will stay for if you use it on soil, the microbe will stay long term into the soil, and it can change for a longer effect the composition of the soil and make it for example, detoxify. One of the effects is it can detoxify heavy metals in the soil. So in that case, we prefer to use the microbe for better effects. But usually we use a mix of metabolites and a microbe because we have noticed that using both together enhance the efficacy of the bacteria.
Okay. Here's a good question. With the microbiome research, do you look at how the bacteriophage populations can modify the diversity and genetics of the bacterial ecosystem? Are you considering bacteriophage
It's not something
effects?
we have done. There's a lot of it's really a vast subject, and we haven't had time to look all the aspects of this subject, but maybe in the future we will do.
Yeah, that's fair. Christina wants to know if you've looked at eukaryotic microbes and their potential impacts.
No, we haven't. We haven't done such studies yet. We are still. We haven't.
Yeah,
We have.
I guess it's
We have been studying bacteria and microbiome for more than 20 years, and there's a lot of still a lot of things to discover and to study.
yeah, that's for sure. All right. Well, that's the end of this time slot. So thank you so much, Celine,
Thank you,
for
Patrick.
coming and chatting with us. It's always good to have a perspective on the microbiome. Now we are at the point in the program where we've got our first scheduled coffee break. It's 20 minutes. So I think this is a good time for everyone to step away, use the bathroom. I'm going to go grab a snack. I'll meet you all back here shortly, and we're going to hear from Sharon about more plant and micro partnerships. So get ready for that. Yeah, thanks everybody. I'll see you in a little bit. Hey there, Luke. Looks like your internet's doing okay.
Yeah, well, I'm on cellular data at the moment, so we'll see how it goes.
Yeah, I'm glad to see that Milton didn't get you too bad.
Yeah, we got pretty lucky in the end, I think. We lost power for a couple hours, but that was it. So,
Wow.
yeah, it's just trying to get the Wi-Fi back now, and they couldn't. We were hoping they could troubleshoot it online, but they're going to have to get a technician to come out this evening. So, it's
I see.
yeah, it's a work in progress, but.
Yeah, if it would be helpful for you, I could like present your slides or something. So you just have to talk. If you're on your phone, that could be tough.
Yeah, I mean, that would be great. If we still have the issue tomorrow, if the technician can't fix it, then I can send my slides over to you, and we can do it that way. That would be great. Thanks.
Cool. Yeah, no problem. Just let me know. All right. So we should probably keep going on schedule. It's 10 a.m. Pacific now, and we're going to hear from Sharon Doty, who's going to tell us about plant microbial partnerships for reduced nutrient and water requirements. Hey, Sharon.
Hi, yeah, let's get that started here. There we go. Okay, I assume you can see that. Yeah, so happy to tell you all about our work on how we can use plant microbial partnerships for reduced nutrient and water requirements. I'm a professor here at the University of Washington in Seattle. Okay, as now we've heard in some of the earlier talks, we can use plant micro partnerships to help promote plant resilience to stresses and to increase nutrient availability. So we want to help plants reduce those stresses that impact our ability to have the sustainable space habitation. So we can tap into the power of endophytes, and those are the microorganisms that are within plants. They can have an incredible impact on plant growth and health. So my lab is, for the last 20 years or so, has been studying all these different ways that the plant microbiome can help plants. So including resistance to plant pathogens, including fusarium, which was pointed out that was one of the pathogens of concern. They can increase plant growth with reduced fertilizer requirements. They can produce plant hormones. They can increase drought tolerance and tolerance to other stresses, and also reduce phytotoxicity of pollutants. As was also shown in the previous talk, it seems like the key is to source these endophyte strains, the microorganisms, from plants that are growing in really stressful environments, so particularly the ones of interest. So I've 3 graduate students. You'll hear from Morgan shortly this afternoon, astrobiology graduate student, and she sourced endophytes from plants growing in arid regions. And Kevin Schaffman, who will give a talk tomorrow. He's also an astrobiology program, samples plants growing in lava fields as well as in these other nutrient-limited environments. Our nitrogen fixing strains that I'll focus on are from these river systems in Washington state that are natural systems fed by high alpine snowmelt, so they're super low in nutrients. So again, I'll just be talking about the nitrogen and water use efficiency. So nitrogen is a key macronutrient that plants require for growth. 80% of the air that we breathe is dinitrogen gas, but it's inaccessible. So only certain bacteria, nitrogen-fixing bacteria, are able to reduce that dinitrogen gas into ammonium. That's basically fertilizer for the plants. But it's been long thought that only plants such as legumes that have these root nodules that house rhizobium can benefit from that symbiosis with nitrogen fixing bacteria, but that's not true. There are lots of non-nodulating plant species that are pioneers that are able to colonize these really bare substrates, like from lava fields and these rocky substrates, glacial retreats. So this is our focus for nitrogen fixing bacteria. We've shown that there is this nitrogen fixing activity occurring in wild poplar is our test subject. We put them in these sealed flasks, dose them with N15 dinitrogen gas, and then they become labeled. So we've used both direct and indirect methods to show that nitrogen fixation is occurring in these non-nodulating wild plants. And it's filled with a lot of diazotrophic or nitrogen fixing bacterial species or genera. There's quite a variety in there. So neat thing about endophytes are that many of them are culturable. So unlike soil bacteria, these microbes that live inside of plants, about 70% of them grow readily in our culture conditions. So we can study them in detail and even add them to other plants. So we've shown that we can add some of these wild microbes to cultivated poplar, and then we get increased greenness, chlorophyll, and total nitrogen. But the neat thing about endophytes are that they have a really broad host range. So we can also add them to grasses, so monocots, which are quite different from the eudicots, diverged millions of years ago. And yet these bacteria are able to colonize and fix nitrogen in all of these different plant species, tomatoes, even conifers. So we've been doing mechanistic studies on these nitrogen fixing bacteria. It's commonly thought that bacteria have to have this anaerobic condition in order to fix nitrogen to protect their nitrogenase, where they need to be in this microaerobic environment for this activity to occur. But many of the bacteria that can fix nitrogen in these poplar plants are aerobic, and they actually have less activity if it's in a microaerobic environment. So kind of breaking some dogmas. So our lab has been working in collaboration with the Pacific Northwest National Labs to study nitrogen fixation in these endophyte strains at the biochemical level and cellular level. So we've shown that just certain subset of the bacteria are fixing nitrogen, and then we're looking at the indicator molecules that are involved. We've found using fluorescent microscopy that some of these nitrogen fixing bacteria are active within these what we term nitrosomes. They're these little compartments within the plant cell. So that was just recently published this year. Okay, so using these nitrogen fixing bacteria, we can help reduce the amount of fertilizer that's required in these space stations and for habitation elsewhere. So if we are going to eventually use regolith, we want to be able to grow the plants in that would not be carting fertilizer all the way to Mars, for example. So being able to access plants, microbes that can just pull that nitrogen from the air and convert it to ammonium would be a great benefit. Okay, so another way that these endophytes can help plants is through water use efficiency. So I think water is going to be greatly limiting in these habitations. You said that most of that water is recycled and then increases in salinity. So there are microbes that will increase salt tolerance. That I'll talk about this reducing the overall needs for water. So with this nitrogen fixation and also antimicrobial compounds, hormone production, those would be really direct mechanisms that are involved. So the microbe can fix nitrogen, they're providing that nutrient. They're producing antimicrobial compounds, they can fight off those plant pathogens. But how they can increase stress tolerance is not known. Stress tolerance of the plant. So one can be a reduction of reactive oxygen species. So we think that might play a role. Also production of phytohormones. So a lot of these microbes can produce things like indole acetic acid, a plant hormone, and abscisic acid that influences stomatal control. So anyway, we can add these a consortium, so usually about 8 to 10 strains of bacteria from the wild plants, and add them to inoculated plants. And then we see this increase in resilience. So in this experiment, the poplar plants had their own microbiome, but it was only if they were receiving these this consortium of microbes from wild plants were they able to survive even a month without any water. So we don't think it's from increased rooting because they're in these small pots. So it's probably hormones and antioxidants and other such mechanisms. So we found that it also helps with even conifers had life or death consequences. This is looking at mortality. So if they were subjected to drought conditions, then the controls 10 out of 10 of the plants died. But if they were given these endophytes from wild poplar and willow, then only one of them died. So a great reduction in mortality under drought conditions. We've studied this a little bit more in other crop plants, so such as rice. So looking at the physiological levels, and found that overall, if they have these bacteria, then it reduces transpiration. So it's kind of closing the stomata more, and so there's less water loss. So we think that they're able to do photosynthesis with the doors shut, basically, because the microbes within are respiring, so metabolizing the sugars and then producing carbon dioxide. So under extreme stress, the plant can still do photosynthesis with the CO2 that the microbes within are providing. So overall, we're seeing this reduced water loss and increased growth. And so overall, water use efficiency of productivity. We've done similar experiments in poplar, and also showed that there's this increased level of photosynthesis and reduced water loss through the reduced stomatal conductance that improves the overall water use efficiency. That was also just recently published this year. So these are nature-based solutions. So we're trying to use the microbiota that wild plants have selected in these challenging environments. So we go to these nutrient-limited environments where plants are just growing in rocks and sand, and sourcing the nitrogen-fixing bacteria from there, for example. So the studies that I just barely touched on here over is research from the last 20 years or so. So from a variety of funding sources, we've licensed out these strains to Intrinsics Bio and also Intrinsics Environmental that really moved all of this research out into the field. So we're grateful for that. So I've touched on, acknowledged some of the people in my lab already, and also wanted to acknowledge the Pacific Northwest National Lab and the Professor Soo-Young Kim's lab for their collaboration doing the plant physiology work. Our current studies into the mechanisms is supported by the Department of Energy. Okay, thank you.
All right, thanks, Sharon. That was really interesting. It's cool to know that my favorite tree, the willow, has some special superpowers I didn't know about.
Yeah, great.
Um, we've got several questions. One of them is from one of our panelists, Luke. He wants to know, he also is going to give a talk about nitrogen uptake
Oh,
tomorrow.
great.
Um, he asked, he'd be interested in hearing your thoughts on the potential for co-cultivation of nitrogen fixing crops like legumes with those that can't fix nitrogen, specifically in the space environment.
Oh, yeah. So that's routinely done on Earth agriculture, that they will do that, or at least as crop rotations. So yes, it's well known that there's some excess nitrogen secreted from the legumes, and so then the non-legumes can benefit. But so but it's great that these non-leguminous plants can also have nitrogen fixing bacteria in them. They wouldn't have to be dependent on that, on partnership. So you could just grow lettuce, for example, without having lettuce next to clover or beans or something.
Okay. Another question we've got is, do you simply isolate the endophytes or do you also propagate them in culture?
We isolate them and grow them in culture. Yeah, so these are all readily grown under normal microbial conditions. So yeah, we can maximize the growth.
Yeah, I guess I have a follow-up question to that, which is I don't work with microbes that much, so I'm not familiar with the protocols. Is it, if you wanted to introduce a culture of these endophytes, do you just apply them on the surface like you might with another microbial inoculant, or do you have to inject them? How does that work?
Yeah, it's very simple. So if it's like a tree, we can just pour some of so it's washed culture, pour that onto the soil, and then it's just taken up by the roots.
Hmm.
But for annual crops, you can coat the seeds with the bacteria. And another way to do it is to do a foliar spray. So we can fluorescently tag these endophyte strains, and then we can track how that colonization is working. So we can use the foliar spray, for example, and then the endophytes still end up in the root systems. So they're entering the vascular system of the plant, and then they are traveling throughout. And we can add them to the roots, and then they propagate throughout into the leaves, for example. So they go through the xylem and phloem, colonizing the plant fully. So it's part of the plant microbiome.
Okay, that's pretty interesting. Ellison wants to know, do you know how poplar or willows perceive the wild endophytes? And do they use a similar mechanism as rhizobia, like the nodes, or
Oh,
how does that?
no, yeah, so no root nodules. Yeah, are very interested to know that early communication of how the plant recognizes them as beneficial microbes and lets them in, because obviously plants do have a strong defense mechanism to block out pathogens. And other studies have shown that those pathogen resistance genes or defense genes are upregulated initially, but then downregulated. So it's like the plant sees, okay, you're a bacteria. Usually the flagella is the trigger
Mhm.
recognition of the flagellar proteins. But then its defense is downregulated, so they're letting them colonize. So somehow they know that these are the good guys. And since it
Interesting.
has such a broad host range, um, from um, including angiosperms and gymnosperms that diverged, millions and millions of years ago. I think that it may be just a very basic symbiosis, like mycorrhizal fungi. So the vast majority of land plants associate with mycorrhizal fungi, for example. And so it may be that it's a similar um, symbiosis, that these endophytic bacteria have been there from the beginning. Yeah.
Yeah,
Recognized
okay.
as normal.
We've got time for one more, I think. Do you suspect that developing a sort of probiotic from the natural nitrogen fixers your group collected could be a useful method to combat microgravity stress?
Yes, and I'll let my grad student Morgan Raimondo tell you about that aspect. Yep, she's
All right,
studying
sure.
stress tolerance.
Thanks. We'll look forward to that then. I think those are all the questions we have, Sharon. Thank you so much for your time and for this presentation. So the next person on the list here is Nicholas Heim, who's supposed to tell us about fungal fuel cells, graphene, and microenergy, which is really interesting. However, I do not see Nicholas online. Nick, are you here somewhere and I can't see you? I don't think so. So in that case, hey, Frida, are you still around with that backup presentation?
Yes, but do you want to go on maybe to the next paper and maybe the first author will show up the next, as you wish.
We can do that, although I like to, in case people are trying to attend specific talks, it's nice to not move the whole schedule forward, So I think if you're okay with it, I'd prefer to just slot you in now.
Okay, yeah, I can. All right, so put my presentation on. Okay.
Thanks so much.
Okay, it's just going to take a moment.
Yeah, no worries.
Did you want your camera on? Yeah. So where's the? Am I on?
Yep, looking
Okay,
good.
you can see the slide.
Mm-hmm.
Okay, well it's good because I'm going to talk about too much nitrogen being bad. So all right, so we're going to work on closed ecological systems, in which most people work on components of systems and figure out that when all the components are put together, that somehow things are going to work out okay. But I'm working on complete systems, and trying for simplicity, we have 3 species of green algae as photosynthesizers. We have Daphnia magna that eat the algae. We have undefined microbes, unfortunately, but we've saved a lot of samples. And we have chemically defined media so that we can change whatever we want. And in general, we keep the phosphorus as 1/12 of the nitrogen, so it should never be limiting in these studies. It's bad enough to deal with the number of variables that we're dealing with. So if you have an open system, and when I say 0 nitrogen, it means that the algal inoculum, which is 10% of the total, has brought in a certain amount of nitrogen with it, but we didn't give it any more nitrogen. And the Daphnia populations did reasonably well. We increased the nitrogen. They did approximately the same. We increased it some more. They did a little bit better. These are in the open systems, and this is sort of what you'd expect. The more nutrient you give, the more algae you get, the more food there is for the grazers. The surprise came when in closed systems, if we put in a lot of nitrogen, the Daphnia drop dead. The algae do very, very well, but the grazer population just dies out. And it's somewhat variable about this amount of nitrogen. It turns out to be really related to the carbon and nitrogen. And we think what the mechanism is, if you have fairly low amounts of nitrogen, the pH increases. So this is dissolved oxygen. The oxygen increases a little bit, the pH increases a little bit, but if you put in more nutrient, so when you open the systems, we lose a certain amount of the gases. but we add more nutrients and then the pH will go up to above 11. And if you look at the way in which bicarbonate adjusts itself, we can go into that detail. This is a well-known CO2 model. The point I want to make is that when we put in lots and lots of nutrient, the algae, if they get a head start, raise the pH to a level that the Daphnia died. That was kind of unexpected. So again, if we're working with fairly high amounts of nitrogen, the Daphnia population may hang in there for a while, but it dies out fairly quickly. Whereas if you have reduced nitrogen and you have increased carbon, the Daphnia population can hang in there. Now, another interesting thing is that when the Daphnia population has lots and lots of food that dies out, the algae do reasonably well. If the Daphnia population does well, it really crops down the algae. Now, I didn't make a major point of the fact that our methodology is such that if we are measuring pH and oxygen and a lot of other variables, we only have one unit that does that, so we can't do any statistics. But if we simply use bottles and put in a pH indicator, then we can get some estimate of pH, and we can look at whether the replicates behave the same way. This was an interesting experiment in which the replicates did not behave the same way. So we had 4 replicates where the Daphnia died out very promptly, and the algae grew up and then its chlorophyll decreased over time. In those in which the Daphnia did not die out completely, the Daphnia managed to reach reasonable numbers and survive for a reasonable amount of time. This is 240-day experiment. So, I mean, the fact that we had the Daphnia last for approximately 100 days is doing pretty well. If you have this L-Splitsville kind of system, it totally throws your statistics into disarray. You simply cannot show statistical differences when you have this kind of variation, which is a problem in replicated experiments. So we've done a lot of manipulation. One of the things I worried about was whether the potassium was too low, and we did an experiment where we increased potassium, and it made no difference whatsoever. In many of these experiments, we're putting dead Daphnia in the algae only, so that all of the nutrients are the same between the Daphnia and algae and just the algae. So we then modified that experiment and added additional nutrients. And where we added additional nutrients, the systems responded very, very, very vigorously. So if we go back, I don't know if I can go back to, one of the things you notice in most of our experiments is that putting the nutrients in, you get a big splurge of algae, you get a big surge of Daphnia, and then the system kind of just degrades down. And we hypothesize that this is due to the fact that we don't have adequate nutrient recycling. And I think you'll notice in the presentations today, people very much are focused on how much can we produce? Can we produce it quickly? Can we put in really high nutrient levels and really high light intensities? And will that make the system more productive? And when you have the whole system together and you don't have the atmospheric contributions of carbon dioxide and the ability to lose oxygen, the systems behave differently. And I think that we need to be aware of this as people work on subsystems, that eventually we're going to have to put the whole system together. It came as sort of a surprise that the carbon, nitrogen, and phosphorus ratios were not the most beneficial in a closed system if you have them the highest. Now, there are other closed ecological systems that have been researched years ago, and I just might point out that there was a blossoming of closed ecological research in the 1950s and 60s, and then it kind of died out and it's kind of being reborn now. The University of Hawaii did closed ecological systems, in which they argued that their systems were essentially perpetually recycling nutrients. And they had experiments that lasted sometimes as long as 7 years. They also, the University of Hawaii studies were the basis of ecospheres, which were sold as sort of decorative desk items where you had shrimp living in little tiny closed systems. And then some of those shrimp lived for 7 or 8 years. So theoretically, or from that point of view, it did appear that they had a system that replenished itself. I cannot say that that is the case for my systems. So I don't know how many minutes I've used, but I just wanted to give you a brief overview of what happens when you try to put all of the component systems together and you look at a total ecosystem as a unity. Thank you.
Thanks for that, Frida. It's always great to hear the latest on the microalgae, or the, I guess, microalgae, Daphnia, mad ecosystems.
Well, let me just say that my first job after I got my PhD, my first job was at the University of Washington to study the feasibility of using human fecal material to grow fish for astronauts.
How'd the fish taste?
The fish tasted fine. We autoclaved them. Part of the grant proposal was that we had to eat the fish. Well, okay,
Gotta
so
put your
it
money
was,
where your mouth is.
well, it was a Boeing sponsored project. They supplied the human fecal material, which had been freeze dried. It looked exactly like the fish food that you would buy in a store. We were going to have microbes break it down, and then we were going to have algae grow, and then we were going to have Daphnia, and then we were going to have fish. And the fish decided the hell with that noise, and they just ate the pellets directly. And it wasn't nutritionally adequate. We had to add additional nutrients. But I think that one of the things you're seeing in this meeting is lots of people are focused on production, relatively little on waste, reusing waste, having a closed ecological system where all of the nutrients have to be recycled.
Yeah, that's definitely one of the large knowledge and technology gaps that we identified a couple of years ago. You're well aware, I'm sure. I had a presentation I gave at the International Conference on Environmental Systems earlier this year about a solution for solid waste treatment using wetland waste treatment systems. But I am not giving a talk on that. I'm not planning to. Although one of our interns at the Spring Institute, Claudia, is giving one tomorrow about something related to that. So we got a couple of questions. There's a pretty good one from Luke here. He said, if pH is the driver and the increase is due to nitrate uptake, have you tried using a combination of ammonium and nitrate as the nitrogen source to balance the pH effects?
We have not, and that would be interesting. The problem that we looked at in the past was that the carbon a lot more carbon is used than nitrogen. And so I don't know if I can. Okay, so the pH seemed to be driven in many of our studies by what's happening to the carbon. The nitrogen is much smaller, the nitrogen transfer, but I appreciate that idea, and I think we ought to test it. There's a big gap between what I think the system should behave like, and my experiments very frequently tell me to go to hell and behave differently.
Yeah.
So
We've
yeah,
talked about
thank you.
some length. I think kind of in the same vein, Dan wants to know about bicarbonates in the system, which I know that's something you think about quite a lot.
Well, we are using bicarbonate, and part of the difficulty with bicarbonate, okay, so this is total carbon, And what happens is the total carbon decreases to about half. But what is happening is the carbonate is coming up, and this is what's responsible for the high pH. So what I'd like to do is switch to using CO2, and we're playing with that.
Mm,
You know, in a life support system for space, your plant growth will help use up the CO2 and provide oxygen. So that'll be part of a total system. But introducing CO2 is a bit of a problem, and we've just recently had caps drilled so that we can inject gases.
nice. Jane has another suggestion for the pH. Have you considered abiotic buffers, like a mineral?
This has been suggested, and we've actually bought some HEPIs, and we've done the literature surveys on what concentrations are non-toxic to Daphnia. Whether excess oxygen, we cannot find any papers on high oxygen effects on Daphnia. And we're hoping to start some studies next week on that. So, but we have planned an experiment using buffers to try to handle the pH problem.
Yeah, in fact, I think I recall a conversation that you and I had when we were trying to puzzle this out, and you were concerned that the nutrients weren't being recycled, particularly the chitin and the carapace of the Daphnia.
Yeah.
And I guess maybe calcium bicarbonate could be one of these mineral buffers that we use to try to impact the pH. But I guess you have to find a way to get that to be bioavailable for the Daphnia. Which I guess is the problem still, isn't it?
Well, I
Because they
think
don't have a lot
the
of.
whole question of nutrient recycling is
Mhm.
important, and I think the European Space Agency with their they have
Melissa.
an anaerobic digester as part of their system.
Hmm.
I suspect that anaerobic processes may be important as well as aerobic processes.
Yeah. Yeah, I guess your systems are all aerobic all the time, huh? Interesting. I know you also experimented with refugia in an effort to maybe keep some of the algae away from the Daphnia in the systems where the algae crashed first, but that didn't have much of an impact either, huh?
No, but there are other experiments in which if we left out the carbon, for example, where the algae tended to cling onto the cell walls, which in a sense protects them from grazing. So, you know, you think you're going to set up a simple experiment that is going to have one variable, and the biology throws extra variables at you.
Yeah, you said that the algae was clinging to the wall of the glass, I guess, but you have 3 identified algae species, right? And none of them are benthic.
Correct. And also
Interesting.
none of them none of them actively swim. And yet in spite of that, they have vertical migration behavior. So that, for example, we're measuring chlorophyll in the manta, where the sensors are coming from on top. The algae actually swim up toward the sensors at night because the chlorophyll sensor is putting out light, and the room is dark.
See. Interesting. Fascinating. Okay. Well, yeah, I think that are there any other questions? We still have a few minutes. Dan recommended maybe you could add chitinase, the enzyme, directly to deal with the chitin if that's a concern.
Well, the only problem there is that we don't want to digest the Daphnia while they're alive.
That's true. I recall you also tried an experiment where you used the substrate, like you just used natural lake water, including some mud, which we
Well,
would.
yeah, we did that, and we did another experiment, which I didn't include here, where we added Lake Washington biota. It did not improve recycling, but
Yeah.
you know, that was one example, one sample, and we have not yet really moved into estimating or doing the DNA on what our microbial populations may be, but I have saved samples. And if we had more complete microbial communities, I mean, my impression of a microbial consortium is that you have a bunch of organisms, each of which uses the waste product of the upstream organism. So organism A puts out a waste product that would otherwise inhibit itself, but organism B is using that as a substrate. And so the whole microbial consortia research is a very complicated research. It's not a matter of just adding individual bacteria one at a time and seeing how they'll do. You really have to add the consortia in order to have it function, I hypothesize.
Mm-hmm. Jane says maybe there are some fungi that can do the chitin degradation for you. I guess there probably are aquatic fungi that do that. They have to, since the whole ocean is full of chitin.
Well,
Okay.
Pseudomonas bacteria are well known for breaking down chitin.
Are they? Okay.
And one would have to look for strains that are non-pathogenic. And of course, most of the research has been done on the pathogenic strains.
Yeah, of course. Well, Frida, thanks again for pinch hitting on this one. I really appreciate that. And it's always fantastic to hear about where the origin of closed ecological system research came from and where we are now. So thanks for this presentation. I'm going to move on to the next talk. It's from Louis Guzman. And yep, all right.
Hello, everyone.
Welcome.
Yeah, thank you for organizing this. It's been a great event so far, so really interesting talks. So should I start sharing my screen?
Yeah, I think you'll be able to override Frida, who stepped away.
All right, so yeah, sorry for I need to present in this mode because if I move into presenting mode, it will probably freeze and make things terrible. So, well, yeah. Well, I want to introduce myself. I'm Luis Guzman. I'm an artist and researcher, currently doing my PhD in Northumbria University as part of the Space IDRT. And I am also a permanent member of the Space and Planetary Exploration Laboratory at the University of Chile, and part of Biofutures for Transplanetary Habitats Hub in Newcastle, where I'm based. So my work, really, I would like to provide some context about it, because all kind of conducts to my practice in space biology. So for a number of years, I have been developing bio installations. I have been understanding life as a plastic medium, and I'm interested in working with life for its capacity of growth, transformation, and adaptation. So this line of work is focused on capturing CO2, so are intended as sculptures that can grow and develop by exchanging CO2 and oxygen with the audience. So, well, this line of work has several iterations. I tested out several materials and forms of growing plants, mosses, fungi, etc. Also looking into different materials like PCL in this case, polycaprolactone, sorry, which is a biopolymer that is also biodegradable. So I was looking into developing structures that could be dissolved over time, and in this process, releasing elements that could be beneficial for the development of biomass. So in 2019, I presented this installation called Cloud Landscape in Edinburgh University, which was also presented as a post-natural ecosystem for space. I was interested into the built environment, but also trying to provide speculative visions about how can we take nature with us once we move out of the planet. So this is me working on it. Well, I tested out several biomaterials. All of it is biodegradable over time. So it's also a combination of biomaterials and geometries. I was also looking into using different geometries to produce more surface area that could create a more organic structure, but also increasing the surface for plants to grow. So this is how it looked. And when I finished this installation, I got an open call from MIT Exploration Initiative to launch a project to the International Space Station. So this was my first experience working with astrobiology. And given the fact that I was already working with photosynthetic organisms, I decided to continue that line of work as part of the Sojourner 2020 payload, which was a payload with the capacity of simulating lunar and Martian gravities. So my project was included into the lunar portion of the project and was titled Bioarchitecture Cosmoecology and consisted in growing marine diatoms of the genus Phaeodactylum tricornutum under lunar gravity conditions. So I was interested into understanding how space technology could help us to transform life. And I was interested in the ontological and the morphological differences of my diatoms compared to their counterparts on Earth. For this project, there was a small problem with the chambers that you see there, because all of them were intended to transport objects, and my project was the only one that included biology and liquids. So I had to make my own chamber, and this was an interesting development because it continued over time. I have been developed other iterations of these also in other space projects. I'm going to talk about that later. And well, the project was launched in March 2020 as part of this CRS-20 mission of SpaceX and NASA. And it was in the Columbus module for 30 days. So after a month of growing and replicating in space, I got several generations of diatoms that could be considered as extraterrestrials. So that was the purpose of the project, was to create life that could be in itself different ontologically from life that we know in our planet. So it was also kind of a reflection about the ecological and the evolutionary implications of space technology and space exploration. So these are electron microscopies of the diatoms once they're returned from space. Interestingly, Pheodactylum tricornutum has the capacity of changing its morphology between 3 different types. So I had the 3 different types in the sample, and also some organisms that appear to be in a very kind of indeterminate state. So yes, for example, this is the triradiate morphology of the diatom. And this is the fusiform. And I have 2 more fusiform samples here. Well, the project was presented digitally in the festival Ars Electronica, looking into speculative visions about space ecology, and also was published in a Antenna Journal of Nature in the Arts. So from the perspective of looking at these diatoms as research, I was really interested also into in situ resource utilization, because diatoms seem really promising for several applications, ranging from oxygen to biofuels to hydrogen to methane to nanoelectronics, because they have these complex silica structures that interact with light. They can be also used for drug delivery, and they are capable of producing pigments and fucoxanthin. They're good for water filtration and can be used as biofertilizers as well, not mentioning also their capacity for CO2 sequestration. So I was thinking, like, if we are going to go to the moon, it could be a good idea to take some of these organisms with us, because, for example, MOXIE, which is the main technology for producing oxygen, it in itself is limited given its capacity, but diatoms replicate once a day, so you can grow them very quickly in these conditions. So this is kind of a map of everything that diatoms can do. Well, and also because, as I showed before, I had to develop my chambers for this project. This got me involved with the Space and Planetary Exploration Laboratory at the University of Chile. So in 2022, I started a series of collaborations with them. And the first of those collaborations was the PlanSat project, which was a CubeSat taking extremophile bacteria from Chile into space. So for that project, I collaborated with astrobiologist Dr. Jenny Blamey, who has a very interesting collection of extremophile bacteria. So in this opportunity, we took bacteria from Antarctica to the Atacama Desert. This is how the project looked like, and this is how it looked integrated in the satellite. So I developed another series of these containers and collaborated with the development of an instrument capable of measuring its growth in microgravity. The 2 images that you can see at the bottom of the slide are actually really terrible, because these are the first photos that we were downloading from the PlanSat once it was launched. Well, the project was launched in the Transporter 4 mission in April 2022. And interestingly, one of the extremophile organisms that were involved in this project is Deinococcus radiodurans, which I thought it was special because it represents one of the most ancient DNAs on Earth. So there's kind of this story about the evolution of life and its expansion in space. So basically, I have been really interested into understanding these organisms as extraterrestrials. So probably if you never met an alien before, well, you probably could consider these as extraterrestrials. So that would be all from me. Thank you so much.
Okay, thanks, Luis. That was really interesting. This is exactly what I was talking about when I said that I'd love to see some cross-disciplinary talks, and it's fantastic to see bio-inspired art going to space as well. Yeah, that's really cool. So I have a quick question while we're waiting for people to put theirs in the Q&A box. PlantSat, really interesting. I hadn't heard of that experiment, although you said that it contained a bunch of extremophile bacteria. Does that mean that there were no plants on PlantSat? Just
At the end, there was no plants because, well, everything, both projects actually were kind of a little bit constrained by the pandemic. So we developed the first one during 2020. And the main project that was the plant going in the CubeSat, the team in charge had faced a few problems. So at the end, the main project was also about bacteria.
yeah, that's very common with space experiments, is making do with what you have.
Yeah, the name is stuck because once you register the CubeSat for your mission, then you got the name and it's really hard to change.
Yeah, that's totally reasonable. Good to know. Christina's got a great question. How do you feel that art influences your research? And do you find creativity essential to your scientific pursuits?
I consider myself as an artist. All of these projects have been presented in the form of installations, prints, projections, etc. So for me, it's more about understanding and collaborating to the construction of the narratives about life in space rather than providing technical knowledge. But some of what I do, because it's experimental in nature, also could serve for other collaborators and colleagues to take some insights from it. So I'm not looking to create a cutting edge science, but I guess that all of these experiences could be nutritious for the community in general.
Yeah, absolutely. All right. Andrew's got a question. Is it typical for one species of diatom to have specifically morphologically different phenotypes?
Well, diatoms are a huge, they have a huge variety. Sorry, my English is too well today, but they can be really diverse. In my case, Phaeodactylum tricornutum is one of the diatoms that doesn't have a hard silica structure. So that is the reason why they can change. But most of maintain their geometry over their lifetime.
I see. Okay. Why did you choose diatoms for your opportunity to send something to space?
Well, because I only had a cubic centimeter to launch something to space. So I thought about microbiology. And I also knew that for me was really important to have something that could experience space flights in a way and be transformed by the conditions of space rather than launching something that would return the same as a it could be, for example, a small piece of sculpture or an object. So I thought like, okay, and I'm also interested into the symbiotic dimension of microorganisms and aerobic organisms, because they have produced the conditions for life in our planet. And I think that is really fundamental to be understood by the wider audience. And so I use these opportunities to share that narrative as well.
Yeah. Yeah. To clarify, does this species of diatom show those same morphologies in normal gravity, or were those unique shapes?
Well, I guess that they show the same morphologies in normal gravity, depending on changes in carbon availability. But there's some diatoms that I found in my sample that look to be in the middle of different morphotypes. So, well, I would have loved to have more images or a way to measuring changes during the experiment, but it was kind of very discrete in terms of what was possible. So
Yeah.
I only have a sample and the sample that was launched and returned.
Gotcha. That makes sense. All right. Yeah, I think that's about the end of our time. Thanks so much, Luis. This is a really
Thank you.
interesting one. Really grateful for your presentation. Next up, we've got Matthew Paddock, and he's going to be telling us about harnessing synthetic biology for biomanufacturing in space, from fermented foods to industrial enzymes. Matt, are you on
Yes,
right?
I'm here.
Oh, fantastic.
Great. Yeah, thanks for the introduction. Let me get my screen shared. Is it all coming through properly?
Yep, hear you loud and clear. We can see your screen.
All right, great. Thank you. So, as Patrick said, my name is Matthew Paddock. I'm here with the Synthetic Biology Project at NASA Ames Research Center, and my talk will be harnessing synthetic biology for biomanufacturing in space, from fermented foods to industrial enzymes. So, before I start, I want to go over the background of what is synthetic biology actually. So the National Human Genome Research Institute has a fairly good definition that I want to use here. Synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. So for the remainder of this talk, I'll be using that definition when I refer to synthetic biology. Another thing I want to mention about biological systems in general and why they are useful for long duration spaceflight and other space related applications is that they're scalable, programmable, precise in that they can produce certain compounds in a very precise manner. They're also the only route of production in some cases. The example I'm using is protein therapeutics, and they can handle some low temperature ranges, and extreme pressures. And as talked to much in this conversation already at this workshop, is they're regenerable and good for closed life support systems. So synthetic biology in space. Mission resources will need to be produced on site for mission sustainability and cost effectiveness. This includes fuels, food and nutrients, chemicals, plastic binders, medicines, and a variety of other compounds. And biomanufacturing can provide compounds abiotic systems cannot. Thus, harnessing synthetic biology for these applications is a very viable way to increase closed life support. The NASA Synthetic Biology project here at Ames is focused on two different experiments. The first one is bionutrients. It's currently a flight experiment. It's in its third iteration flown on the ISS. And the goal is to develop and demonstrate an on demand nutrient production system for long duration missions to mitigate nutrient degradation in stored food. So far, we just came out with actually the Bionutrients 2 and Bionutrients 3 patch designs, as you can see below. But in essence, it's a project in which we have a dried media and a dried organism, and we rehydrate those in space and incubate them to produce a product that can be consumed, in theory, by astronauts in the future. The second one is the CO2 based manufacturing. It's developing and demonstrating a prototype system to enable microbial manufacturing using abiotic CO2 conversion systems to products that drive biomanufacturing for long duration space missions. So it's a slightly different take on synthetic biology and using abiotic systems as a way to help improve the efficiency of these systems. So the bionutrients elements, as I said, there's three different ones that we've had so far. Bionutrients 1 actually launched in 2019. On the right, you can see some pictures from the initial bioreactors on the space station, actually when the experiments were getting run. But it's a 5-year flight test using engineered yeast strains to produce carotenoids, hence the orangish color you see there. It was also to test media and related packaging to see how well these things will store and ship, and also to test a Gen Zero bioreactor design, which, as you can see, is this kind of cylindrical style. It was also to look at different microorganisms for long duration storage. So we have a variety of organisms and organism storage conditions to see how they're going to store in long duration space flight. Bionutrients 2 was launched in 2022, and it was a little bit of a shorter mission. And it was hoping to expand what we had. Originally, we were just growing yeast in kind of a nutrient broth, but we are expanding that then to producing yogurt and kefir, which are more palatable options of microbial fermentation. We also wanted to engineer the yeast to produce other things, such as medical countermeasure products. And we also tested a new bioreactor design. So on the right, you can see the different bioreactor design. It's more of a bag in this case, a gas permeable bag, and it allows, well, less mass for one, but also more efficiency in that we can have more bags in the smaller system. We also wanted to develop a HACCP food safety plan so that we could start looking at these for use for consumption. And one thing I want to mention, so on the left is the different yeast strains, and there's a variety of different colors based on the products they're producing, which will be more clear in the next slide. And on the right is the yogurt. And one interesting thing that was developed for the yogurt was a pH indicator um, to see how quickly and efficiently it actually is fermenting the food, because yogurt needs a certain acidic pH in order to be consumable. So in this case, the blue color will turn to a yellowish color at the end of fermentation, indicating that the organisms have properly been fermented. For Bionutrients 3, this is an upcoming flight project in 2025. We're looking at engineering a yeast strain to produce multiple nutrients as opposed to just a single nutrient at once. Looking at a Gen 2 bioreactor, which is a slight take on the bags that we're currently doing. And we also wanted to do more food related stuff, so demonstrating the reliability of our HACCP food safety plan using an eNOS for pathogen detection and pasteurization. And on the right, you can see the bags, and there's a slightly different pH indicator we've decided to use that's also edible, so it's further allowing these systems to be used in space. So for BN2, I want to go a little more in depth of the products we looked at because a lot of these were things that we had to engineer using synthetic biology, which is the basis of my talk. So we used a variety of different organisms related to food. Saccharomyces cerevisiae was the main one we used for zeaxanthin production, and it was hoping to be a way to mitigate eye damage as a nutrient as well. And you can see on the right the different colors of yeast we have, all indicating the different products they're producing. And then we had Saccharomyces boulardii, which is actually a subspecies of Cerevisiae. It's a probiotic yeast, but we were using that for beta carotene production for vitamin A, which helps with eye-related Uh, the next yeast we looked at was Cluvermycins lactis, which is a dairy yeast, and we engineered that to produce fulstatin to counteract different effects of microgravity. And then in terms of yogurt and kefir, we just started looking at the different bacteria there. So we had Streptococcus thermophilus, producing GFP just as a demonstration that we can engineer the organism. And then we had yogurt, which was a commercial yo mix we used, and we had Cefir, which is a commercial kefir mix. And in all these cases, like I said earlier, it's a dry mix of organisms and the substrate they're using, and in space they just rehydrate them and then incubate them to produce the products. So here's some fun pictures of the Bionutrients 2 operations. No leaks, no anomalies, and the visible growth. I like the picture on the top right to show the extreme color variations between the different products we're producing. And as well, left of that, you can see the initial blue color of the yogurt. And on the bottom, you can see it turning to yellow. So similar to Joya's talk, the astronauts seem to be happy and enjoying it. That might have just been for the camera. I can't say for sure. The next experiment I want to talk about is CO2-based manufacturing. And this one's hitting on a slightly different aspect, but very similar to the aspects of bionutrients, in that bioproducts are difficult to store and often need to be made on demand. Commercial production systems are large, bulky, and not optimized for use on other planets other than Earth. We need to utilize local resources via in situ resource utilization. So the ConOps of our project is we would use these rapid CO2 conversion systems that can take CO2 directly and convert them into carbon sources, typically salts like formic acid, formate, methanol, ethanol, acetate, and then use these as substrates to feed into biomanufacturing platforms using much more readily engineered organisms. And then we could use these to produce bioproducts, which have a variety of applications. So on the left is kind of this is kind of the overall um, design of how we were going about this project. We have the CO2 conversion systems. Here at Ames, we had one that we've been using to make formic acid. We also partnered with Stanford for an acetate conversion system. And there was also the NASA Centennial Challenge, where we were challenging companies to find a pathway from CO2 to glucose using purely abiotic means. Not shown here are also methanol and ethanol, which have recently been commercialized from CO2 conversion systems. So that is something we have more recently looked into. And obviously, these CO2 conversion systems would have their carbon sources fed into the bioreactor design, which is the CO2 based manufacturing platform, which would grow the organisms, harvest, and purify the products. We also looked at a variety of different organisms and different genetic tools, and all of these in the end would be applied towards, long duration spaceflight. So our current use scenario is we're looking at acetate and ethanol as carbon sources, easily produced from CO2 conversion systems. And we're using the pretty typical recombinant organisms, E. coli and K-Phosphy, having them produce a carbonic anhydrase, and then we also have a K-Phosphy engineered to produce cutinase. Um, and these would be important for the carbonic anhydrase to potentially be used for CO2 removal in liquid amine systems. And cutinase is a nice enzyme because it's very highly secreted in K-Phosphy, but also useful for plant biomass and potential plastic degradation. So here's some early pictures of the platform on the right, the prototype platform. It was designed as a semi-autonomous system to grow, harvest, dewater, lyse, and purify the recombinant proteins from microbial cells. We also looked at developing a gravity-independent high gas exchange membrane bioreactor to improve microbial growth. And this platform overall demonstrated validity of the concepts and identified challenges for future investigation. We have a nice gel on the bottom, just showing the different stages of K-Phosphy secreting cutinase, in this case, going through the system. And in this case, we were able to not only separate out the cutinase, but we showed a really high level of concentration in the elution, which was very positive for us and shows demonstration of an entire small scale platform for this sort of synthetic biology application. I want to thank the synthetic biology team, all the current members and past members for their hard work in this project. We've been doing a lot of hard work and quite a few interesting things we've come about with the CO2 based manufacturing and bionutrients projects overall. Um, and it's been quite a team to try to manage, especially with the bionutrients, having the flight experiments going on and making sure that we can coordinate and get reliable data back and forth. Well, that's the end of my talk. Thank you. Are there any questions?
Wow, that was really something, Matthew. Is quite overwhelming at how advanced you guys are. It's pretty impressive to see. You've already got 2 flight experiments under your belt. Um, I guess while we're waiting for the questions to come in, I've got a couple. Of course, there's the obvious one. Anytime someone is making food, you have to ask, have you tasted your yogurt and other products?
I don't think I can answer that question.
All right, fair enough. Well, my follow-up is that I want to know if you so I guess I was inspired by seeing your blue yogurt. And of course, I doubt that it tastes like blueberry quite yet, but I'm curious if you looked into other microbes capable of adding organoleptics or something to improve the flavor profile of the yogurts and other products you're making.
Yeah, flavor profile is definitely something that we've talked about. We're not quite at that point of developing it, but that will be an important one if we decide to move forward and have this be consumed because um, yeah, there is really not much flavor going on. It's a rehydrated milk powder that's fermented, so it's
Mm,
not going to have a lot of flavor compared to commercial yogurt production. But yeah, I mean that's definitely something to consider down the road. Is how could we make this more palatable and tasty as well? Because as mentioned previously, flavor and taste tend to be diminished in space. So trying to get it so the astronauts receive as much benefit for this as possible.
right. Uh, Kevin has a question, which is, did you notice any changes in microbial fermentation in microgravity, like genetic, transcription, physical, etc.?
Yeah, that's actually a great question. We're actually analyzing the data now from a lot of these experiments to see how things performed. In most cases with these, we had a set time period, so it would be, the yeast would grow them for a set amount of hours, harvest, and just look at the final products. In most cases, the ground controls and flight experiments, things were able to get fully through fermentation, and we reached the products. But in terms of how the organisms react and how they look and how they respond in space, that's still something we're trying to comb through the data and get better information about. But yeah, I assume there will be some differences just because the environments they are very different.
Hmm. You mentioned briefly about methane, I think. So these were all CO2 based, like derivative compounds that you fed. Do you have any, just, I guess, like how would this change if we were going to use methane as a feedstock? Because I think that's a waste stream right now on the ISS.
Yeah, so if we were to use methane, we would run it through some sort of conversion system to make it into a solid compound. And there are a few out there because, yeah, any sort of gas ends up being an issue, especially as some of the plant people talked about, you don't have convection and mass transfer changes in microgravity. Um, so there are systems to convert methane into larger chain compounds, and that would probably be the only way we would use it. I know there are companies out there actually using methane in bioreactors currently for biomanufacturing, and that's been a challenge just dealing with that mass transfer because methane does not like to dissolve. So
Yeah. Okay. Any more questions from the audience? Oh yeah, okay. Dan's got one. Um, other than nitrogen use efficiency, how does nitrogen look for this approach? Wait, Dan, can you clarify that? Oh, here's a link to a paper that might shed some light on that question. Nitrogen accountancy in space agriculture. That is an interesting title for a paper, but I'm not positive what the content of the question is. Oh, as in
I think
source.
I think I
What
have
is that?
some sense, and I can try to answer it. I guess for both projects, the bionutrients one, we're shipping all the materials up there, so it's not ISRU or a closed system in that regard. We're just shipping things up there, nutrients that would otherwise be degraded in fresh foods that they otherwise wouldn't really be having access to. Um, with CO2 based manufacturing, nitrogen comes up as a really interesting point because if we want to use in situ resource utilization, finding alternative sources of nitrogen is important because after carbon, nitrogen is kind of the biggest compound microbes in use. Um, so we've looked at some other ways. There are abiotic sources of nitrogen or ways of converting nitrogen gas. There are also organisms that can do it. And then we also are looking at different waste sources, like if we could just recycle nitrogen sources from urine or other waste products, that could potentially increase the amount of ISRU we can accomplish with this system. I hope that touched on it, but I know exactly the paper. When you said there was a paper, I was thinking exactly of the nitrogen accounting one. So it's a very good paper that talks a little bit about accounting for nitrogen in these instances.
Hmm. All right. Ted's got a follow up. What kind of return protocols do you use for your ISS experiments? Are they returned in cold stowage?
Yeah, we return them in cold stowage, and look at them. Basically, from there, we have them stored immediately, and at the end of the experiment, we have them frozen away and shipped down. That way, we can analyze them without really changing the conditions too much.
Hmm. Okay, that's a great answer, short and sweet. Um, do I see? Yeah, one more question from Kevin. For this system on the moon, how do you deal with nitrogen in situ resource utilization? As I understand, there's virtually no nitrogen or hydrogen on the moon.
Yeah, no, that's a very good question. The only real way we could deal with it is recycling waste we brought with us. I think the larger application was for Mars because Mars does have nitrogen. But yeah, if we were to use it on the moon, which in theory is the gateway to testing a system like this on Mars, we would have to recycle wastes or do something else because the moon really doesn't have much nitrogen with it. And, it's not going to be a source of a lot of nutrients in general just because it's pretty desolate.
Hmm. Yeah, that's right. Okay. I don't see any other questions, so we're coming up to the lunch break that's on the schedule. It's going to be an hour and 10 minutes, so we're going to reconvene here in 12:30 Pacific time. Um, I'm going to leave this Zoom webinar running. You guys are free to stay connected or to drop out and come back in. The same link should work either way. I'm going to pop in periodically just to check on things and if there are any questions or issues people are experiencing to help troubleshoot those. But I guess, guys, thanks for sticking with us through the first 3.5 hours of this. I'm looking forward to the second half of the day. We've got some more really nice talks. If you can't make the second half, of course, don't worry about it. It'll be recorded. So, yeah, I guess I'll see you all in an hour and 10 minutes. Thanks, everybody.
Patrick,
Yeah, what's up, Oscar?
how much time do I have? I'm living an hour here to figure out in Florida. How much time do I have?
Uh, I had trouble hearing your
Oops.
question, but I think you're asking about the duration of your presentation.
It's coming in and out.
Yeah.
Maybe
Can
you
you
can
type the question?
text me. I will try.
Yeah.
Give me a second here.
Well, now I think
Try
I
to
can hear
find.
you better.
Okay, yeah. Just how much time do I have for the presentation?
It should be around 15 minutes, so there's a little 5 minutes for Q&A.
Yeah, that's okay. So 15 for presentation and 5 for Q&A. All right.
Yep.
Yeah.
Patrick, I have a question. I can see the questions that are in the question and answer, but I haven't been able to enter a question in the question and answer.
Yeah, we realized that the
Oh.
people that are on the panelists, or basically all of the speakers, are not able to submit questions because they're technically able to respond via text in the Q&A box to questions that don't get asked out loud. I've just had Kevin, for example, has just been asking me the questions via direct message, or you could just throw them in the general chat. I'm watching that too.
Yeah, I wasn't sure that you had time to look at those while you were handling things on live. And have you heard from Christine?
I got an email from her earlier today. Apparently, she woke up feeling really sick.
Oh.
So there's a chance that she might not be here for her presentation this afternoon. If that's the case, I do have a recorded presentation from Gabrielle Caswell, who's actually our last talk today. But the video is quite long, so it might be appropriate to play it during the featured speaker spot anyway.
Okay.
Yeah, that's my
Alright.
plan right now, if Christine
Alright.
doesn't.
Okay, I'm going for lunch.
Yep, me too. Thanks for your talk today, Frida.
Okay.
Patrick, are you there?
Hey, Oscar, how's it going?
Good. I'm gonna try to share
I'm having trouble hearing you still.
the way, and and then I'll unshare it, and that okay might be a problem, huh? Can you hear me now?
I
No?
I can hear you a little bit better, yeah.
How about
I saw that
now?
you just sent me your presentation.
Yeah. Can you hear me? No. Uh-oh. I think we're hung up, huh?
Oscar, if you're having issues with the internet, you could try dialing into the call through the phone instead of computer. And then I might be able to share your slides for you.
Hey, Patrick, can you hear me now?
Yeah, now I can hear you much better.
Okay, I'm gonna try to share again. Hopefully, just wave if you can't hear me.
Sure, yeah.
I'm just practicing here.
Colin can also hear you.
Okay.
Sounds crystal clear. Yeah. Thanks, Oscar. Actually, Pat, how do I sound then? I'm sitting here in a train station.
Yeah, no, you're good too.
Awesome. Cool.
Yeah. Thanks for being flexible with the presentation there, Colin.
My pleasure.
Oscar, have not seeing anything if you're still trying to share. Just a couple of minutes left until we're resuming the program. I see people are logging back into the call now. Hey, Davi, welcome. It's nice to hear from you. Are you on today or tomorrow? Uh, Looks like tomorrow, I think. Wait. Um, yeah. Yes, I found it in the program. All right, well, it's 12:30, so we should get started again to make sure we stay on track. Um, Ideally, we were supposed to be hearing from Christine Escobar, since Joya very kindly took her spot earlier today. I did get an email from Christine. It seems that she woke up today feeling very, very sick, and I know she was traveling also. So it seemed like she might not be here, unfortunately. Uh, However, we do have some other people that are able to step in, and so I think that this won't be a big interruption. We're just going off script a little bit. Colin has volunteered to take this first one, and I think we will just go ahead with Colin, and then we'll see how the rest of the afternoon unfolds after that. So Colin is going to be presenting a really interesting talk. I think he's going to be talking about mapping metabolic triplets, visualizing self-organizing principles based on Gibbs free energy and an ever-adapting ecology. Take it away, Colin.
Okay, how's the vocals, and can you see the presentation?
You sound good and also look good, so.
Oh, hey, I try. You know, it's not easy being this cheesy. Okay, well, hey, thank you everybody for having me out. Once again, shout out to Dr. Dan. Hi, Dr. Frida, and of course, Dr. Christina. Patrick, thank you very much for the time. So mapping metabolic triplets, visualizing self-organizing principles based on Gibbs-free energy and ever-adapting ecology. What? Okay, I'm talking about wetlands in a box as bioreactors, essentially. Once again, my name is Colin Lennox. I'm the principal for Eco Islands LLC, and I've been working on self-organizing wetland bioreactors, SOBs, or wetlands in a box, for about 15 years now. Most of my clients are coal mining companies, so I've had a lot of exposure in a good way and a bad way, one could say, to a lot of different mining reclamation impacts. Iron, aluminum, and manganese are some of the big ones. Sulfate, selenium are most of the ones that I deal with. I live in central Pennsylvania. And we're starting to branch out. Like I'm here in Grand Junction right now, Colorado. Just got done working at the Mars Desert Research Station on a refit crew, and now I'm going back to keep working on a wastewater bioreactor, a wastewater soap specifically for Arcology X in Montana. So I'm actively building terrestrial systems. But yeah, so wetlands in a box. So just what is a wetland? We spend a lot of time talking about individual microbes, and my concern is often that we miss the forest for the trees. When it's one thing to be able to adapt through CRISPR a microbe, but when we know very little about the wetland ecology and the concentrations and diversity of biofilm within a certain environment, feels like, well, why don't we look at that, all of the above, not one, not necessarily the other to exclusion, but it helps to know something a little bit more about the native environments here on the planet. Because I think sometimes we're, for the sense of simplicity, we're missing the diversity and then the easy, low-hanging fruit, the wins that are represented in more diverse ecologies. So that's my take on it. We need it all. We're going to need it all. But anyway, so just briefly, and to those who've seen some portions of this, there is one new slide. That's the important part. But just to go over some basics. So what is a wetland? Basically, it holds water most of the time. Water moves through it. High surface area to volume. High biomass to volume. It cycles matter and energy in lots of ways, biotic and abiotic means. And it self-organizes by Gibbs free energy and the prevailing atmosphere. I'm breathing oxygen. So are you. That's our prevailing atmosphere. But for methanogens, no oxygen. They are obligate microbes. If there was oxygen present, it would kill them. So that's why it's so important to know what is on the, what's above the water or outside the sphere, so to speak, within a zero gravity environment. And a little Bee Gees reference down there just for fun. We're all just staying alive, trying to. Okay, so and then what is a bioreactor? So if that's what a wetland is, what's a bioreactor? It does predictable and reproducible work, and these are my definitions then. But these are useful for me to then understand and visualize how then to build reactor systems and know at what point different materials may be remediated, removed, captured, whatever term you like. But the biological engine ruled by the laws of conservation of mass and energy. Okay, makes sense. Catalyzes reactions, producing useful byproducts and biological in situ resource utilization. That's why we're all here. Make as much as we can, recycle everything as possible for when we go up. Now, natural wetlands are self-selecting and organizing, and another term is natural attenuation. I like that one too, so I threw it in there. Also constructed, or then in juxtaposition to that, constructed or manufactured wetlands are the conscious selection of environments to cycle matter and energy. So I build manufactured wetlands for my clients as opposed to constructed wetlands, which are generally the norm for wastewater treatment facilities and in particular mine drainage treatment systems. Differences being my systems are usually measured in cubic meters versus an acre of square feet of wetland out in the middle of a mountain area in Appalachia, or even here in Colorado. Okay, and then what is biofilm? Once again, there's a lot of concentration on individual microbes. It's important. We have to know what the individuals are doing, but then also when we go making a lot of changes in the genetics of microbes without the understanding of their role in the larger ecosystem, that's something that needs to be better elucidated. Yes, both, all. I'm all for it. But here's the low-hanging fruit is the way I look at it here on Earth. So what is biofilm? It mixes microbes and their secreted colloids. That's that EPS, the extra polysaccharidal matrix. Biofilm remains generally stable in mass, population diversity, and density. Generally, but there's changes, and that's what we'll be talking more about. Now this EPS, then the film, that stuff on your teeth in the morning, all biofilm. And I'm probably preaching to the choir, but we gotta establish the ground rules there. Protects the microbes producing the film. And then main component in natural cycling. Without microbes, there would be, all those carcasses just still sitting on the ground. If it weren't for them, there would be a lot of there just would be no life, continuity to today. The main components in natural cycling, some of the obvious benefits of biofilm are media for chemical signaling, structural support, refuge, desiccation protection, and the biofilm and that EPS host complex redox reactions. Okay. So the redox ladders. So basically, what I work with is, if anybody's familiar with the Winogradsky columns, I build big Winogradsky columns, and instead of them being on a classroom table standing up and down on a vertical glass column, ours are many cubic meters in size. They're horizontal, and then they can be sealed or left open to the atmosphere. Here in this case, Earth's atmosphere. Once again, going back to that, what is the prevailing atmosphere? So the redox ladder there, and I've got to go ahead back and find where I pulled that one graph from, just so I can properly cite it. I've been using it for a while, but it's the basic concept of, you got oxygen at the top, and if you think about ATP being produced through the metabolic triplets of oxygen, and in our case, since we're chemoheterotrophs, the food we eat is the energy source and the carbon source. One of the reasons we're so darn efficient and we have these large bodies to begin with is because our metabolism is so darn efficient. And it's 32 ATP being produced. I'm pretty sure that was Krebs cycle. And then on the other side of that coin is methanogens. Generation of methane through carbon dioxide reduction only yields 4 ATP. It's a very inefficient process, but it's good enough. These critters have been doing that thing since long before the Great Oxidation. So perhaps some of the very earliest microbes on that ever existed on our world, we could find up on Mars as well. I think the evidence that is presented is, at least in my mind, it passes the smell test, no pun intended. But just to keep in mind then, so in the field, a very simple $30 probe, and I consider myself a wetland ecologist. I'm a field guy. I'm not a lab guy. But an ORP probe measures in millivolts, and an oxic system or aerobic system is plus 450. Anything above zero, really, if you think of it as a number line. And then negative 450 millivolts, generally speaking, is methanogenic, which means you've got 0.9 volts in between those 2 points. So if you hook a Winogradsky column up and it's oxic at the top and methanic at the bottom, you'll read about 0.9 volts under best circumstances. I mean, there's always exceptions and rules and the like, but just generally speaking. And between all those different points then is the amount of ATP that's being produced, or the Gibbs free energy is another way to look at that, delta V over something or other, I forget. I'd have to go back and take another look. But the idea is generally that, after oxygen, what is reduced first? Nitrate. So your nitrifiers. Nitrate provides quite a lot of energy. It also volatilizes, so it's very easy to lose to N2 within a sealed atmosphere. So maintaining that nitrate as nitrate that is usable is difficult. So you got to catch it when it's ammonia, essentially, to be able to oxidize it to nitrate. Anyway, long story short, you can see that there's those multiple steps: nitrogen and the manganese, iron. Selenium isn't in there, but it has a higher ORP than sulfate, and then the methanogens. So you get the idea. It's stepwise, and it can be skipped if as a wetland is changing and adapting and remediating, it removes all the oxygen, it removes all the nitrogen, and it goes down through those individual steps until you get down to methanogenesis. Assuming there are enough nutrients still available within the water source. There's ways that you can stop the entire thing cold, but just I'm speaking in broad generalities here. These are generally how wetlands work here on Earth. Okay, and once again, so there's a Winogradsky column. Just an idea that if you take a mixed sludge and put it into, mix it up, put it into that Winogradsky column, and then give it some time, it will then begin to self-organize based off of if you've got open atmosphere oxygen at the top and you've got, say, methanogens or sulfate reducers or sulfide reducers down towards the bottom, you could see it in visual layers. And this is what Dad taught me. He was a microbiologist, rest his soul. But these are some of the things that he had taught me. Very easy to see visual representations as learning tools. Okay, now for within the scope and the context of for chemists and engineers, because a lot of times they're just scratching their heads whenever I'm talking to them. So I just throw this slide in there just for in this context and applications. SOBs, or self-organizing wetland bioreactors, are multi-fuel, multi-oxidant biological engines self-organized to use each reductant and oxidizer sequentially based on the energy released and availability of the metabolic pathways. If you don't have those microbes in there, you could skip over it and it wouldn't happen at all. So this is why it's important. And it would be impossible to go up without taking most of those microbes with us anyway. So it's one of those ones where, you know, I worry about, like, you know, are we going to lose certain metabolic pathways when we go up? Probably not, because we're taking them with us. We rely on them to survive in the first place as these big multicellular organisms. And that metabolites can be skipped over if the genetics aren't present. And that in that case, it may just leave that metabolite or, you know, that contaminant within the wetland stream, and that will then pass through the entire system. Okay, there we go. And of course, you know, other ways to look at this is in your terminal electron acceptors, your terminal electron donors, and of course your carbon sources. So those are the three primary metabolites. Let's see. Okay, so just these are further units that I've built by hand myself. This is at one of our mining site, removing aluminum and manganese. And once again, most of my research is in manganese oxidation from solution. I'm trying to figure out when it happens biologically and when it doesn't. We're still, the jury is still out on the specific conditions, but we think we're narrowing them down. So that's my primary line of research. I've been at it for about 10 years and a lot yet to learn. Now, I won't go too deep into this one. Several of you have seen this before, where this is a theoretical best conditions for a regolith processing system. This one is on Mars. This was presented to Mars Society years ago, where the pink units there are regolith different processing systems. The purple are different salts. The green over there are the habitats. But the important part, and at least in regards to this presentation, are the blue ones. And that is pretty much the same lineup as that step-based one that I showed you just a couple moments ago of the redox ladder. So you would have a series of reactors, assuming you had all of those, a heterogeneous waste stream with all of those material in it that is then capable of, that also has all the genetics available in the microbes for full remediation of that system. Of course, salt passes right through any system, as in standard wetlands. It's easier to get the water out of the salt than the salt out of the water. By salt, I mean sodium chloride, potassium phosphorus, your halides and your lanthanides primarily, which is cool because those actually end up being your chemical plants. So knowing the difference between what can and cannot be remediated with wetlands or just in general is very important, because, you know, you can do a bunch of the metals, but you can't do the salts. But simple electrolysis will do the salts. That's how we get all of our table salt, chloralkali process. Simple, easy to understand, requires energy and carbon electrodes. So that's, you know, keep it simple. Stupid. Okay, we got just a couple more of the reactors there that I built, just a different viewpoints. By the way, that's aluminum that is precipitating there. Aluminum is not part of a metabolic process or a metabolite that I am at least personally aware of, generally speaking. But just wanted to point out that, you know, biofilm still grows within the context and uses attachment surface area. Now, whether it's indirect or direct bioremediation is another talk. But my final slide here, now this is, we're going to be publishing this in the Journal of Mine Water, and Reclamation, I think. We presented this at Task Force Mine Drainage. conference in West Virginia. But the basic idea here is that so depending on the pH, and understanding that certain microbes, whether they be archaea or bacteria, will then adapt their environment for stable pH and then kind of keep themselves there, sort of like a holding pattern, and remediate, in this case, iron under low pH conditions down to about 2.6 is the lowest that I've seen on any of my sites. It'll go lower than that, but I've seen 2.6. And so you get your iron first, and then you would go through different processes. So, okay, after that, how do you get your pH up? Sulfate reduction, potentially. Drive off hydrogen sulfide, and you've got alkalinity left over. Drive up your pH then, as you know, balance of your hydroxyl and hydronium ions, and then you get that up to 4.5, and then aluminum. And then, and if you've got enough residual alkalinity, then you can get out all your iron as well. Now, those iron and aluminum oxidation are also acid-producing reactions. So you have to have enough alkalinity to buffer all of that to get yourself up to about a 6.5. So depending on what pH you need to get to is how much you would drive off. Then you got your manganese, and usually that's about a 7. So this is that visual representation I was talking about. So that is actually my time, I believe. So I will go ahead and kick over to questions. But the big idea, when considering Gibbs free energy and what the different redox steps are, gives you the opportunity to say, okay, I know where I am and where are we going. And that's by and large the point of this system then, and in general how Winogradsky columns work. Maybe time for one question, and thank you very much for your time.
Thanks a bunch, Colin. I always love your chat because it's such a great representation of, literally ecology, checking out the interaction between the microbes and the environment. And it's good to bring some of that biogeochemistry into the conversation. Something
I love it. Yep.
Sorely lacking.
Yeah.
If you guys have questions, go ahead and put them in the chat or in the Q&A box. Yeah. It's okay to go a little bit over time here because we're kind of going off the script. I've got a question for you, Colin. When are you gonna try using lunar regolith simulant as the substrate in your SOABs?
Yeah, just presented for Johns Hopkins LSIC here the other week. That's a good question. And right now, my really, I think it was Livermore just asked us to do a return to Glasgow. So really, my focus is mostly on sulfate reduction. Now, if there's a basalt lunar regolith simulant, I'd be happy to. But that's the problem with lab scale versus getting out into the field is the representation of all those different, ecological components. And so that's why I sort of stayed away from it. I'd love to, but I think I'd probably fail. Is maybe what I'm saying. I'm not sure that I've thought about it. Yes, whether it would work or not, that remains to be seen. I'm just as interested as leaching for chlorate and seeing what that would do, because that would deliver us Mars results. Mm-hmm. And then you've also got fuel, and that's ClO4, so that's a lot of oxygen to work with. I'd love to work on it, but is there enough? The amount of regolith I would have, when you consider, some of these mine drainage sites are pumping out, 100 mil of iron at 100 gallons a minute. How do you replicate something like that in a lab-based system? When if the microbes that are being engendered are concentration related, where there has to be enough energy? Because that's the thing, is like as you're removing one, there's a fall off and then a chance for something else to take hold. Mm-hmm. It isn't necessarily just immediate stop. Okay, we're at 0. We used it all up and we're done. It's okay. Now there's not quite enough left, and now you have a new metabolic advantage for the next metabolic step. Yeah. Can that be achieved or overcome in something like lunar regolith? That's one of those ones where I'm just going to say that's a great question. I'd love to know when, but I got to stay on my craft with rare earth elements, manganese, and sulfate reduction. So sulfate reduction is arguably more important to yield the totality of knowing you have to go backwards and forwards between oxidative and reduced environments in order to remove or capture and for processing, further processing all of the metals that might be actually in that material, short of having to use a whole bunch of caustic soda or a lot of sulfuric acid for further leaching, which is how I think I would start off the process after you finally ground, leach it, drive off the salts, and then whatever's left, put of the metals, get that into solution, and then see what microbes take hold and watch for what oxides being produced. Sorry, this is a really long-winded answer to I'm not sure, and it'll probably be a while. Let me do some of the other basic work first. But thank you, great question.
Sure. Well, this is an excellent example of some biological institute resource utilization. I didn't see your picture of the big, like 99% pure manganese nodule that you get out of this thing when you're done.
Oh yeah, I do have. Well, I still have a couple of those buckets of was it hexavalent or triclinic bonacite, I believe, from one of our mine drainage treatment sites. We're going to be going back to that site as soon as we can because we're still trying to figure out whether that is biotic or whether it's abiotic. Where we just happen to have found a really rare situation of exceedingly high removal of manganese. And whether I personally, I think it is there is a microbe, manganese oxidizers involved, but that remains to be seen. So, but thank you very much.
Okay. All right. So thanks a bunch, Colin. That was really excellent as always. Again, we're off script a bit. I think what I'm going to do now is we have a video, a pre-recorded presentation from one of our colleagues in Australia. Typically, we don't do pre-recorded videos for this conference just because we're trying to maintain some interactiveness with the presenters. But it's really hard to join if you live in Australia. So we made an exception. And this video is a bit longer than a typical speaker slot. So maybe it's a good thing that that we've got some time from a featured speaker slot to use. So I'm going to share the screen on this other computer. And okay, I'm just going to play this video from Dr. Gabriel Caswell. And I guess I'll also put her email in the general chat so that you guys can follow up with any questions directly to her. Okay, and without further, well, I guess actually let's make sure that you guys can hear it. Can you hear that? Because
Hello.
Hey,
Hello.
Oscar.
Hi. Did I miss it?
No, you haven't missed anything yet.
Okay, if you run it, I'll just talk it because I was trying to share and it wouldn't work, too low a bandwidth, and I could just zoom through it. Or am I still bad?
No, we can hear you just fine. I think that your presentation is not quite yet, but
okay, good. I'm here. I just wanted to let you know.
Yeah, no, I appreciate that. Yeah, we've gone out of order a little bit. No, okay, so we can't hear the audio from this video. Let me just mess around on Zoom really quick and see if there's share sound. There you go. And optimize for video sharing. That seems relevant.
Or disease sort of.
Okay, nice. Here we go.
Good afternoon. I'm Dr. Gabriel Caswell, and good morning, good afternoon, wherever you might be. I'm going to present just a small sort of, I guess, cross-fertilization concept of humans and their ecology. And the subtitle's on us, in us, and around us, because humans, of course, are part of the broader ecology. There's a little bit about me. I'm a clinician in northern New South Wales, inland on the outback, very rural, very remote. I still work in my clinics, and I am the proprietor of Spaceport Australia, which is an independent research institute. My particular area fascination, really sort of molecules and microgravity, but through that we've sort of strayed into the macro and microbiome area and trying to sort of, I guess, start the conversation to move away from space medicine or a disease sort of concept of space and medicine into one of human health and survivability in space. And I have no declarations of interest, and my email address is on this page if anybody so desires to communicate. Humans and their ecology. So humans I consider a bit of an active universe. There's sort of their own little pocket universe, so to speak, and we have on us and in us and around us a very personalised ecology, which contributes greatly to our health and our welfare. And coming to light in various clinical sort of scenarios and axes, as medicine likes to call them, seems to be this symbiotic relationship that occurs between the microbiome that inhabit every human and the physical and cognitive health of that human. And I think medicine may use some of the terminology just slightly different to what the ecologists do. So I thought I'd just give you how we would use them, so to speak. So we would consider the biome as the Earth's biomass, basically everything from the soil upwards, so to speak. The macrobiome is basically what we consider the immediate habitat, the room or the surroundings of a human, and the microbiome is the bacterium and everything else that live on us and in us, so to speak. And you'll find in some papers it's generally referred to as the genes, as it's in toto sort of encapsulated term, which I think sort of is a little bit misleading, so to speak, but it's a convention that seems to have come out of somewhere. Now we know that the human microbiome is made up of sort of bacteria, viruses, and all sorts of other species that inhabit the human body. We know that they outnumber the human genes by about 100-fold in the genetic material that they carry. And they've been with us for so long, they're like an old pair of slippers. And I think that they have, over time, become very functional and have very broad influence on our human physiological regulation. And in some cases, you'll find them being sort of discussed as another bodily organ. And that sort of, I guess, gives their status of how integral they are to sort of human functioning. Now, the microbiome on humans interacts dynamically with its host and its local environment. And it's important to note that the competition varies remarkably over time and between individuals and between different ages and the stages of life. And it can also be influenced by things like diet and the broader human macrobiome environment. So clinically, we're just beginning to understand how this might all go together, and that, you know, the microorganisms which assemble and, you know, in early life, and if we have any disruption of that, then we are going to get problems with that human down the road or as they grow up. And we know that in pregnancy there is a massive drive and a change happens, and there's a mammoth increase in the diversity in a pregnant woman's microbiome. And after birth, obviously that is transferred to the infant, and basically it's you know, done by breastfeeding, by touching, by introducing the baby into the community that they live in, and this naturally will stimulate its own neonatal gut microbiome. So if we interrupt any of this and we end up with something that's perhaps not ideal in the gut, we know now that it is a progenesis for asthma, for immune depressions, and we know that it might change the nutrition uptake in children, leading to childhood obesity and type 1 diabetes in later life. Then we have links to the adult metabolic syndrome, which is sort of like an endocrine disruption syndrome. And then basically we have what we call a microbiome dysregulation with the obesity. And then there's an issue that we might get cognitive decline or no cognitive development to its full potential. Now, there was a twin study, obviously I think everybody remembers this, where one twin went up in space and one stayed on Earth. It is in the paper and I have referenced it at the end of the talk, but in the twin study it was a little snippet where basically there was quite a bit of cognitive decline that had not recovered after 6 months back on Earth from the spacefaring twin. And I have a very erudite colleague and he said, you know, we spent an awful lot of money learning that n is equal to 1. In other words, there's been very little comment about this outcome or its implication, and I think that this is part of why we need to start cross-fertilizing with other disciplines, particularly the space medical people, because we really need to move towards space health, and this is obviously a problem that is flagged and not solved. So we don't walk alone. We acquire our microbiome first from our mother and our immediate family, and then we basically can have even that section sort of or part of our microbiome life cycle interrupted with a C-section or bottle feeding or antibiotics, and particularly in Western countries this is quite a thing with older women, IVF births and multiple births generally as a result of IVF. So I think that the lack of acquisition, you know, we go back and we have a look at what it sort of increases the risk of metabolic immune disease, and I'm going to throw it out there, probably emotional and cognitive developmental delays, and I think that's probably an interruption of what we would term the gastrointestinal brain axis. So as humans grow, it's becoming more and more obvious that what they eat, what they wear, what they put on their skin, their cleaning agents, their medications, and modern life itself is beginning to impact on the microbiome balance. And clinically, this will contribute to a dysbiosis of their gut, their skin, their cognition, and their just general well-being or healthiness. And I think that we have to sort of look at some of the linkages, maybe, again, yet unproven, but things like disease and organ systems that are sort of subpar functioning, and how the nutrition and what the microbiome does with us needs to be teased out to see if there is some sort of disease sort of fighting mechanism in there. But clinically, thus far, there might be more, they seem to pop up every day, but we have the gastrointestinal brain, skin, immune and lung axis. And obviously the gastrointestinal lung axis is one that I find, you know, in general public team to know about. If they have a very bad gastroenteritis, you can just about put 10 bucks on it, as we say, and within a month they'll end up with a chest infection. And I think that, you know, it's probably a little bit more sophisticated than just the gastrointestinal skin axis, for example. I'd say there's a few more steps in there that are yet to be teased out. But as we do more research and we continue to sort of broaden our knowledge base, I think those interconnections will be identified. Now, the individual human microbiome, so everybody who would be listening, is now believed to be selected and maintained as a result of your personal genetics, environment and nutrition. And I think environment and nutrition everybody sort of understands, but the fact that your genetics may also be working with the microbiome for maximum sort of uptake and health is probably an area that hasn't been explored too much, but I think may, from a space point of view, be very important in the future. So we get changes when we go to space and we take humans out of their natural habitat and out of their personal ecology, and we put them on a spacecraft, we whack them into these imagined habitats or enclosed environments. Analog studies have produced a lot of information in this area, and what we actually start to get is a change, and we get a change in that the microbiome becomes generic with the group. And back in 2016, Voorhees did actually flag this as a challenge for long duration space missions or flights on habitations. And it was more recently looked at by CUNAST as well. And I think that it's important that we put things into perspective, because the microbiota undergoes bacterial or behavioral changes in microgravity. And it's not going to be the same for every species, but we have very limited data. And we know that some of them, like the staph epidermis, is going to actually replicate 4 to 6 times faster than it does on Earth, because we've got the data. But one of the things which hasn't caused a lot of comment, and from my sort of molecular point of view, I guess I look at things like that, is that it has a huge change in morphology. But with that change in morphology, it has a change in behavior. And the behavior is a striking change, where once it's a happy, happy camper amongst all the rest of the microbiome and a very good neighbour, it suddenly becomes very cytotoxic to its neighbours, and it becomes quite aggressive and locally competitive. So I guess my question there, is that a response to microgravity, or has the microgravity forced upon a genetic change? And I guess the sort of the money shot is that change permanent? Because this particular bacteria lives on our skin and lowers the pH of our skin. It lives off the fatty acids of our sebum. And one of the benefits of that is that our enzymes that make the barrier on our skin operate much, much more effectively in a low acid environment. So I think that, you know, these little interconnections, as we call them, are going to have to be teased out, because if this is a particular bacterium that suddenly becomes one of the bad guys, so to speak, then it may be one that we have to somehow manage in our sort of space health programs. So we have other problems, okay? Antibiotics in space are basically useless, and it's probably multifactorial. I would say that we have increased replication times, so antibiotics are taken up by bacteria in a certain stage of their replication for maximum effectiveness. They may miss that window because the replication is just so fast. We have other issues such as fluid and tissue dynamics in microgravity. And then we have the sort of change in genetic sort of morphology that might change the medication that you need to treat a certain type of bacteria. And then, of course, we have traditional resistance, and it might be a very rapid transition amongst the bacteria because, again, we have a very fast replication rate. And I think that, you know, one of the things that has really not been explored is, you know, the fluid dynamics of bacteria in microgravity. Some people have started to look at it, but I think how they move around in microgravity, I think given the change in morphology and the change in behavior, might be quite important, particularly if they become a destructive bacteria to humans. So we're now talking about crewed journeys to Mars, habitats on the Moon, and the Gateway project. So I guess what we need to really start to think about from a clinical point of view is, you know, what is a healthy microbiome on Earth? And what we do to maintain that, we do without thinking. We touch things, we put food in our mouth, we have intimate relationships, we're swapping and gathering microbiome all the time. And it's a very dynamic and it's a very interactive thing. But what we probably don't appreciate is just how adaptive our microbiome are, because our microbiome might initially join our body and settle down, but then we might change our diet. We might go, say, from carnivore to vegan, and basically the gut will change its microbiome over the course of a couple of weeks to suit the new diet. And that's because the nutrients need to be gathered differently from when you're a carnivore to when you're actually, you know, perhaps being a vegan. So I think what we now need to start to consider in the sort of human health type thing is looking at what we call the microbiome homeostasis. And this is not a new concept, but it's not one that for some reason I think has had its rightful place in space exploration, and I think maybe because it's sort of not right up there in Western medicine, and I think it needs to be. But when we're looking at the microbiome homeostasis, we have data from Skylab 1971, where we know the diversity of the astronauts decreased, and they thought at the time maybe it was the heavily filtered water and the sterile food options, but it might also be that they weren't in a macrobiome habitat that was supporting their microbiome. So obviously there's a big interaction externally and internally, and that the transmission from the spacecraft to humans does occur, but of course it's not making up for what we call the Earth norm exposure to diversity. So spacecraft and space surfaces, studies have shown that we have a change in morphology, behavior, and the survival of the microbiota, but would that necessarily be good for the humans in that environment? And I think this is where, particularly if we're looking at Mars and Mars habitats, we really need to bring in technically a supportive ecology. So we know that there is a generalization of the microflora, and the downstream effect is nutritional. So there are some elements, for example vitamin D, that you could become quite nutritionally deficient in or deplete, and that has ramifications throughout all body systems, from immune to cognition to skin health. Then we actually have issues maybe with medication take-up, and it could be added to with fluid distribution, and then the immune dysfunction, which happens incredibly quickly in microgravity or even in LEO. But I think what's important to recognize here is that if we are becoming very general in our microflora in a small enclosed habitat, one person may, for example, be perfectly healthy. The other person may start to become nutritionally challenged. And then we need to look at the hazards the external hazards that may actually affect the microbiome. So once we have some sort of microbiome dysbiosis, we may end up getting metabolic changes. And once we get those metabolic changes and metabolic changes in the human gut, then we might actually get a change in diversity just because the gut environment's changed, a change in the genetic expression, one because of microgravity plus the local environment. We already know we get a dysfunctionality in the immune system, but what the question has not been asked is that actually in response to a change in the gut microbiota. So we have change in diets and we have medications, and then we might need to start to look at perhaps some probiotic therapy, but we have issues with that as well, and human expression, human genetic expression. So at the moment we have absolutely no data on this interplay, which again I think is very complex, sort of simple but complex and complex but simple, but the outcome, whatever it is going to be quite poor for the poor astronaut in that situation. And of course then we have all the other space hazards, as we like to sort of talk about them, and galactic cosmic radiation, that great big Santa sack full of nasties that come on the other side of the Van Allen belt. At the moment the Van Allen belt is protecting the ISS, so therefore we have no data at all. What happens if we pop anything up there for maybe 6 months, 12 months, 5 years? And then we're going to have to look at, human exposure, the time and the length of traveling, say from, the Earth to Mars. And that's probably going to dictate how long they can stay on a certain planet or in a certain environment. And then we have to sort of look at the total radiation account. But when we do this, and though there's very, very limited data, we don't consider the microbiota. And I think this is something that has to be considered, because if we think of it as another organ that contributes to the health and maintenance of the body, we need to understand how it's actually going to be affected. And given that the cells we know are affected in Leo, which is protected from galactic cosmic radiation, then the effect of microgravity plus GCR might be something that is worth considering. And then we actually have a limited ability in these enclosed environments to introduce new or balance the micro sort of biome. And we have to start considering how the microbiome will adapt. I mean, they're very adaptable little creatures. They're showing that very much so in the limited data that we have. But then with that adaptation, do we have a change in pathogenesis? And then we've got to start thinking about the infective potential of different microbes. And of course, we've got no data that enables us to predict anything or extrapolate. And of course, when we look at emergence and complexity theory, there's very little we could probably predict until we have, I guess, enough experience to know that this is likely to happen in sort of 90% of the cases. But then in that sort of no data vault, we don't have anything that tells us which bacteria, if we stick to bacteria alone, are going to become dominant and beneficial. So there might be a microbiological change and that bacteria for some reason becomes more beneficial to us. But on the same token, we don't have any data for which one might become dominant and detrimental. So with the absolutely no data sort of situation, we're not really able to predict or surmise what extended long-haul spaceflight will do for human health. We've got a fairly good idea, say, perhaps, of the microgravity consequences, but we don't have very much concept of what's going to happen to the innate balance within the human and how these things are going to affect, or these changes are going to affect health and welfare and working life. And most importantly, particularly if we're looking at establishing colonies on, say, Mars, you know, we have absolutely no data on different age groups or the stages of human life. So if we move to the concept of microbiome disruption, okay, we need to start thinking about how longer term space environments disrupt it. The research field is open, and the best that we can do is predict from what the limited data we have at Leo, but we're going to have to start to consider how different levels of altered gravity also affect things. So it's not just the microgravity that we might be experiencing in Leo. We're going to have to think, well, Mars is sort of, you know, 50-odd% of Earth. You know, we're going to have to start to think about all the different levels of microgravity, and they may individually affect the bacterium in different ways because it's a different level of microgravity. And we don't have any collective effects. In other words, we have analog data, and we have the world's biggest analog, I think, is going ahead in Finland at the moment, and we need more collective effects. But even that data in an analog is in 9.8 meters per squared gravity. And I think it's probably fair enough to say about this time in the talk, when we start thinking about these downstream problems, that Houston, we really truly do have a problem. So the next thing that I would think about on the sort of space flight surgeon's sort of manual is, well, how do you prepare your astronauts and your people for space, for long-haul space flights and missions? Do we need to start some pre, post and during screening? And then what do you choose to screen? Because remember, if they're up in space, they've got to be able to replicate whatever screening we've done on Earth for it to be meaningful, or for it to be a clinical management tool. And we also need to broaden some of what we're thinking about. It's not just the microbiota changing, it's the impact on nutrition and the availability of proteins and carbohydrates and fats, and how they may be changed if the digestion sort of is changed in the sort of gastrointestinal tract. And we may come to the conclusion that some individuals, for whatever genetics they've got, might be better suited to a restricted environment and restricted diet and space. So that brings us to, I guess, the concept of biological compatibility and not just biological compatibility from a perspective of the microbiome, but it's are there a subset of humans that are biological sort of better suited for space, and do we put people with a high biological compatibility for space together on the same crew so that the crew as a whole is stronger? And this is the thing. Should it be a thing? It I think is something that has not been considered because, again, our sort of knowledge of the microbiome is pretty limited. But then, if we start to look at the individual or the personal astronaut, do we start to tailor a lot of medications and, wellness for that one individual? Do we make a particular probiotic prescription just for them? And then if we do that, how do we package that for space? Because there's an enormous amount of problems with packaging and keeping things in good condition for when it's used. And of course, we don't know whether the bacteria, even if they're put into some sort of stasis, change or genetically mutate, or something happens in flight before they're used 2 years down the track. And again, we get back to this issue: how do we cater for the ages and stages of life? I mean, are we seriously considering things like pregnancy, infanthood, and things like this, which have very different nutritional needs, and are always accompanied by a change in microbiota? How do we start to, I guess, prepare is perhaps the word I'm looking for space inhabitants of all ages? Now, I didn't actually go too much into the macrobiome, but basically we still have no firm data. We have a few hints. We know that if we go outside and have a lovely walk through the forest, our immune system actually gets a good boost. But we don't really understand basically how that external influences works, other than breathing something in and getting into our lungs or our gut. And something that's not often spoken about, and I guess it's one of those things that as we've moved away from more organic materials like wood and sort of fibre, most of our habitats and spacecrafts are made out of plastics and steels and things like that. And that may well actually have a huge effect on how the microbiome behaves itself in space, and may have a huge effect on how the microbiome then evolve, and the implications for health need to be considered. And the reason that I bring that up is that there have been a few flow dynamics studies done on microbiome in space. They're very few and far between, but it's quite interesting to watch their behaviour in from a bacteria's point of view, their different environment. So I guess in summary, we have humans really very much embedded in their biomass, their microbiome and their macrobiome. And all of it sort of works together and contributes to their health and welfare, as well as their mental development. And in elder age, obviously, this all works to help maintain cognition. There is a paper that I saw very recently, very fresh. I think it might have even been a grant that was given out, looking at Alzheimer's and the microbiome. So the sort of, I guess, avenues for research are starting to open up with a little bit more imagination. But we have to remember that all our humans who be travelling in space and colonising are going to be living in exotic or non-natural habitats. And we're going to have a personal effect on the microbiome, the makeup, the distribution in the bowel, the phenotypes of their microbiome. And no doubt there will be health consequences that may be small, might be great, and maybe at a different age catastrophic. But I think we really need to sort of bring the sciences together. We need to start asking the questions: what's going to be beneficial, or what is beneficial may not be beneficial in the long term? Will the external environment of the microbiome become more important in the maintenance of human health? So rather than we focus on the microbiome, maybe it's what we put into the habitats and the environment, and perhaps the little garden in the habitat is more important in human health than we've given it credit for. And will humans themselves adapt their physiology to the changes in their microbiome? And if this occurs, will it be beneficial? Will this be something that we strive to get a better adapted human for the environment that they're living in? And I think these are sort of themes that are sort of a bit right out there, but at the same time, I think the evidence is in the literature of how important the sort of microbiome and macrobiome are to human health. And of course, if we have a longer flights and longer habitation in more remote areas, we also have to factor in altered gravity, and we have to start looking at novel solutions, or perhaps solutions that they can do themselves in those environments, because once they're far away, it becomes further and more difficult basically to provide them, say, a top-up of their personally profiled supplemental microflora. And that brings us to the question of, if we were able to develop this super supplement that had all the microbiota in it that you needed, and you took that once a month to keep everything chipper, what ones do we pick? Because I think the evidence is showing us that regardless of what species you pick, you can expect a change in their phenotype and their behavior over time in altered gravity. So again, we get back to that situation that we don't really understand the function of individual bacteria species, if we stick to bacteria for the conversation, and we don't understand what they contribute. But if we pick the wrong one, and somehow the wrong one changes or doesn't do the job, it could lead to catastrophic outcomes in a remote location. And I guess then we're going to have to look at, or try and imagine, or try and work out how we manage these types of scenarios, because the conversation's just not been had. And we've got to look at, we've got health and welfare, but we also have cognition, which is such a finely tuned sort of organ, our brain, to our gut, that if we don't have the micro elements, it just simply doesn't work. So therefore, we've got to start looking at, I think, how we develop, good, healthy habitats, perhaps. And I think this is where the habitat ecologists, as I like to call them, need to come in and have a say on what we're going to do and how we're going to do it. Because I think human health may well rest in the hands of the ecologist. So I put a few references here for everybody to have a look at. I haven't gone overboard. There is a volume of literature, but it's quite interesting that it doesn't really tell us too much at the end of the day, except that we've got a problem, Houston. And one that's really quite interesting, I think, is the Voorhees in 2016. So he was one of the original that started to sort of flag this as an issue. I think you'll find that quite interesting. And then, of course, we have the more speculative ones about actually looking at crude, crude long haul sort of flights, and the establishment of habitats in off-world locations. Well, I'd like to say I hope that you have found that interesting, provoked a few bits of research, and hopefully some cross-fertilisation of ideas on one health idea that I think is going to be a very large problem if we're going to succeed in the off-Earth world. And thank you very much again for having me.
All right. Okay. Wow. That was fantastic, actually. That was a really, really nice talk by Dr. Caswell. I'm kind of bummed that she's not here to answer questions, actually. I really liked that closing line where she said that human health may very well be in the hands of ecologists. Somebody gets it. Thanks for saying it out loud, Dr. Coswell. Um, Fortunately, I think this has actually put us right back on track, because at 1:35, which is coming up here in a couple of minutes, that's when Oscar is supposed to be starting his talk. So, Oscar, are you dialed in right now, or are you on a computer that seems to be working?
Um, Actually, I decided that I don't have enough bandwidth to be able to share or load. So if you can run the slides and if you can hear me perfectly, we'll get on.
Yeah, we can hear you great. So let me, I've got your slides right here. Uh, Although it's in a PDF format. Is there, okay, yeah, this should be fine. Yes. Okay, I'm gonna share screen again. And all right, I'll go on your cue.
Okay, well, thank you. My name's Oscar Monhe. I'm a plant physiologist. Boy, that's a tough act to follow discussion. But what I wanted to talk to you about is how to, how to increase the number of N for all these microbiome and other studies to be conducted. Next slide, please. If you make it bigger, I think full screen, it will go full screen. Anyway, I'm going to talk about space farming. We always talk about space farming, but I'd like to remind everybody that we are living in a planet. So every farmer that we know, they're already space farmers. We are in space. And so, some of the things that we have to deal with for many thousands and hundreds of years. Here's a couple of times that I borrowed from Bruce Bugby that he gave in a talk on YouTube. Basically, on Earth, light is provided by the sun. The nutrients are provided by the soil and some animal components, a la Martian. And then, you get water from rainfall. Now, uh, as as populations have increased, we've started using fossil fuels for fertilizers, for moving, for expanding, replacing cows. We're using groundwater, and our yields have increased correspondingly, as you can see in the little green arrows. One of the big things that people don't talk about is how much solar energy Earth is really providing us. And Bruce has made his calculations as he always does, and he's calculated that it's about $1 billion per hectare for 100-day growing season. So that's, you know, so when we talk about vertical farms, the cost of electricity is surfacing and it's an issue, but also you got to provide the fertilizer and you also have to have the cost for removing the water vapor, the HVAC systems that also depend on electricity. So what this gets to me is if we're going to have space farms, we're going to need energy for lighting, HVAC, and we must implement some sort of nutrient recycling. Next slide, please. So space farming enables exploration, and then I'm trying to show you here that we live in a gravity well, and there's a if you look at the energy expenditure to get off the planet in terms of a delta V, that's an acceleration by a rocket. We're in a very deep gravity well, not so much on the moon and a little bit more on Mars. And so if if we look at things in terms of these gravity wells, we start noticing that gravity becomes a variable. So some of the things that we've learned at zero G, where there, that uh, it the lack of gravity affects the balance between the capillary and gravitational forces. So when there's no gravitational forces, capillary forces dominate. And if we look at a soil, the bottom one being a soil in 1G, all the water is ponded, like in Even in Florida, it rained a lot and the water's gone because gravity took it away. But if you didn't do that in space, the water, there's no gravity, so it's just going to remain there. And we've seen a lot of plant stress in space because of, this moisture redistribution effects. Other effects are boundary layer driven convection. Hot air does not rise in microgravity because there's no density gradient to move. And that's a considerable amount of convection that goes on and helps mix everything. So without going into detail, if you don't have a fan on you, you're not going to get enough oxygen, or you're going to deplete it. Your oxygen is going to pile up. You're going to get all hot and sweaty. So you got to have a fan on you. And if your fan fails and you're a plant and you don't shut off the lights, you're going to burn up. Anyway, these are all things that we've learned over all these flight experiments that people have been conducting. So the goal for space farming is to adapt our 1G agricultural knowledge into controlled environment agriculture in partial G. Next slide, please. Okay, so how does space farming enable a cislunar economy? I borrowed this chart here from the United Space Alliance, ULA, and it shows a cislunar 1000 economy, and it shows that right now there's like 10, 13 people in space right now around Leo, but it says in 5 years we may have 20, in 15 years we may have 300 people, in 30 years we may have 1,000 people. And what are they going to be doing? Well, they're going to be doing research, right? Or we're going to have tourism or space manufacturing, and then we're preparing to go mining on the moon, maybe go to some asteroids. We're going to be making fuels to fuel all this. We may be having robotics. So I'm not even going to go what's going to happen in 30 years, but a big one is asteroid mining. What if somebody brings back a whole platinum asteroid full of platinum? Big bucks. Anyway, what's going to happen with an increasing population in space? We're not going to be able to feed them sack lunches, forever. And so we got to start growing our crops, and we need new bioregenerative life support concepts to be demonstrated to sustain large cruise sizes in cislunar space. Now, Metzger et al., argue against this, and they say, well, if you're going to, it's too costly to have humans, and so you might have robotics, so okay, but you might still reduce the crew size if you bring robots, but you're still going to have humans in space, and especially if they're going to be living in a moon village or something like that way in the future. Next slide. Okay, so space exploration is also enabled by space farming, and I'm talking in terms of the food systems. The food system must be sustainable, meaning you got to minimize the logistics of resupply, especially as the distance from Earth increases. And so, we've got to provide resources, and I have light in red because of the cost of providing that: CO2, oxygen, the nutrients, water, seeds. We got to have plant chambers and a lot of controlled environment agriculture, CEA knowledge to be able to do this. As Joya mentioned, the plants bring great physiological well-being to the crew. You get that green smells and tastes and flavors, and also, we're finding out that different cultures, depending on where the astronauts are from, they like different crops. So, anybody's eating Mizuna, but Russians sure love Mizuna. And this is just from watching what they were growing in on Mir. So the food systems, they got to provide tasty, nutritious, and safe food and a source of fresh crops, and especially in long duration missions. And here I show some of the plant chamber studies, and as Joya has also mentioned. But one of the things that I really look is how far the Earth looks from the moon, and we plan to go beyond that. So it's going to be scary going out there. So you want to have something familiar. Next slide. So I mentioned the concept of bioregenerative life support. So many scales of plants in ecology, right? So you start with a single plant at the left. It's a pepper plant, and then you got to have some technology to be able to grow it in systems like the veggie and APH. But you're not going to feed a lot of people with zero point 0.2 square meters, so you have to increase and learn how to scale up your production. And this is an example of biomass production chamber at Kennedy Space Center that's demonstrating these technologies. And then you integrate much larger systems than a 20 square meter biomass production chamber into a lunar palace. And then eventually you implement a whole set of crew and how they live and tend their crops in a biosphere too. And then we're doing that in on Earth, and we could be graded on how well we're doing. And then I'm going to mention I've highlighted physiology, technology, and demonstration because those are going to be my next slides. Next slide, please. The first this slide shows it's an eye chart, and it shows control environment agriculture. The goal is, at least in my view, is to optimize crop yield. So I have one single equation here where yield depends on this eye absorbed is the light intensity is the crop. It's converted into moles of carbon per mole of photon. It's got an efficiency for proteins. You gotta respire some of that carbon to make the stuff of life. And then you got a harvest index. Not everything is edible, and you can control your absorbed the amount you absorb by collecting density, and you can change your harvest index by crop selection. And so in spacecraft, we have limited mass, power, and volume. But compared to field agriculture, we can run multiple crops per year. For example, 28 days cycles for lettuce. We can grow 12 lettuce crops, 6 wheat crops, 4 soybean crops in a year. We can use elevated CO2. You get larger plants, reduce transpiration, but you might get also reduced nutrition. You got to condense the transpired water, and then we can, in controlled environment agriculture, you can provide constant light, humidity, moisture, CO2, and you can, spectral quality, etc. One of the things we've learned is this graph in blue over here. It shows that the area required for maintaining a person decreases as the light goes up. So does your cost. But how does doing things in microgravity change that? Well, it depends on the quantum yield for photosynthesis, and we've seen that does not change. So that graph may still be valid in space. Next chart, please. So this next chart shows some of the technology development that's been going on for several decades, and it's been happening. And NASA funded. Now there's other entities that ESA, the Chinese Space Agency, et cetera, et cetera, et cetera. But in terms of NASA, we have they were funding universities, and they're still funding universities. There's work at the NASA centers like Ames at Kennedy and Johnson. We saw Matthew give a description of the work they're doing, and there's tons of small companies that are always doing things. And so a couple of things. I was mentioning sometime in the mid 90s to 2000 that there was near growth, collaboration between the Russians and the U.S. on Mir growing wheat. It's a lot of chambers like the PPS, and then the advanced plant habitat, the veggie, LEDs. Sometime in the mid 1990s, LEDs have spawned a whole vertical farming industry. But that was all started here by a work funded by NASA, and we keep going to the right. You can see here we've been growing space space peppers in space, and lettuces, and we want to beyond. Next slide, please. Hello. Next slide, please. Yeah. Okay. So, can you hear me?
Yeah, go ahead,
No?
Oscar.
Sorry. I'm in my car, so we're fighting technology after a big event here on the planet. Food security might be also challenged. But anyway, so a lot of the technology I wanted to give a big shout out to the biomass production chamber, 20 square meters at Kennedy Space Center. It's an example of biogenerative life support, how plants recycle CO2, and potentially they could also do gray water to produce food, oxygen, and potable water for a crew. It was probably the first vertical farm. And on the bottom, you see how, Joya mentioned that there's several facilities that are new ones, old ones, and current ones, and future ones that will be, doing research on how do plants grow in space. Next slide, please. So I'm going really fast because I wanted to get to the point I wanted to make, that in biogenetic life support systems of the future, and we got to go towards this. We got to recycle our waste biomass. I mentioned that the energy costs of bringing stuff up to the moon or an orbit. And basically, you reduce the logistics of resupply to cislunar space or Mars or the moon by recycling the waste biomass you bring. So if you're able to recover some of the nutrients, you don't have to bring sacks of fertilizer, which I think it's 90 kilograms per person per year, to keep them fed. That was a number that Ray Wheeler came up with. And so future missions on permanent bases, wherever they may be, the moon or Mars, they're going to require the development of biogenerative life support systems. But I added the word hybrid because before it was only the plants will do it. But there's physical chemical systems that are simpler, easier to maintain. They've been tried on the space stations. So we want to combine these technologies to help reduce the requirement for supplies from Earth. Next slide, please. So here's an example of a hybrid physical, chemical, and biological chart, and it's showing how you can close the carbon loop for inedible biomass can be converted into soil amendments. Let me walk through you this. The plants in green, they produce oxygen for the crew, they remove CO2 from the crew, but so do physical chemical systems, and for example, here on the far right, this little arrow that goes to VOC, let's see, human sweat, acetone, and ammonia and other gases, and those have to be removed by the trace contaminant control system, the TCCS down here on the bottom. But that one requires resupply of activated carbon. Every 5 years, you need 50 pounds for a crew of 4. But where's that activated carbon going to come? So one way to do this is, not all the crops are edible. I mentioned the harvest index. So for example, between if you're a lettuce, okay, you eat most of it, but if you're a wheat, you're not going to be eating the stems. So that inedible biomass, you can convert it into biochar, pretty much like people have been doing in the Amazon for I don't know how long, thousands of years potentially, and but that biochar, you could use it as a soil amendment eventually, or to mix with to give you some organic matter. Or you can also turn it into activated carbon, and that could help you again with your physical chemical logistics of physical chemical life support. And then once you have the used activated carbon full of ammonia, full of the acetones and the ammonias, you can use that as a soil amendment again. So this is an idea that I've been working on for quite a while, and how to recycle nutrients from waste that you find in spacecraft. Next slide, please. So this is another example of recycling. You have humans, and we all do two. I'm not going to talk about number two, but we have a lot of urine, and this is one way to do it. I knew Matthew was working with us here a while back, but the urine could be fed to algae because they can handle the high ammonia levels, and then they could produce help remove ammonia from the water, and that water could be used for growing plants downstream. But also the algae can produce oils and proteins and fuels, nutrients, or other feedstocks for other processes, as well as providing some of the CO2. So you can um, start looking at algal strains operating at high light levels. They recycle urine or the urea in the urine for producing fertilizers. You remove the ammonia, and the organic loads from the urine produce nitrate, with nitrifiers, and help with water recovery and contribute to CO2 reduction. So these are recycling processes that could be done with these systems. Next slide, please. And then, this is a simplified chart where, again, you have inedible biomass and you want to recycle it to reduce costs. And then we could do this in space, but you could also apply it to the Earth to reduce the cost of global food systems. So you got CO2 and light by plant and algae. You got the edible biomass, and we can eat all that. But the inedible biomass, it could be used as feedstocks for fish meal, for insect meal, for mushrooms. And then, you get fish protein, insect protein, and mushroom protein, which is edible. And then you got the waste from all those, and you can biochar, compost them, recover some of the nutrients, and go on this cycle again. So, uh, um, this is a way of utilizing recycle waste to make more food, more variety of food. It reduces production costs of protein production, recycles byproducts to recover nutrients for the algae to grow back again. Next slide, please. And this has been looked at, and this is a actual publication by Cowan et al. And where they're using this integrated CEA systems to enable a circular economy. Here they combine it with renewable energy, and you can see how the lines are labeled, but essentially they're creating feedstocks for insects, fish, mushrooms, etc. And it all ends up for our food systems. So you reduce water use for food production, you reduce environmental damage due to conventional farming because it's circular, uh, reduce greenhouse gases, et cetera, et cetera. But mainly you increase food security and environmental sustainability of a global food system with these ideas. Next slide, please. So this is my last slide, and what I wanted to point out is uh, there's many issues to be resolved. Where are we going to put the space farm? Uh, there's a lot of issues with just putting on the surface of the moon, the day length, radiation. Is it going to be underground? How hard is that going to be? Here's another chart from Bruce Bugby, where he says if you're going to use uh, 2 acres of solar panels to produce the energy for 1 acre of production, supply the light. So is the energy source for these food productions to be solar? Are we going to be able to have nuclear? Will fission be developed? These are way things in the future. And again, the question has been asked, can robotics help reduce the human footprint? And then this would be, it would help reduce the crew, which in turn means smaller farms, reduced energy demands, and it may make things doable in a shorter time, potentially. And then how are we going to scale current crop systems for large scale production? Well, we have a growing vertical farming industry that, all the advancements they're making, earth-based vertical farms can be implemented possibly in a plug and play in the future. And with that, next slide. I'm open for questions.
All right. Thank you so much, Oscar. That was a really fascinating talk. Thanks for giving us the overview of bliss and its history and present and future things. Unfortunately, we're a little bit over time, and so I don't want to cut too much into the next person's presentation, but I think if there are questions, people can ask them in the box and maybe you can follow up. But I want to see the talk from Dawn next. I think Donald Kuhn is supposed to be telling us about gas exchange, yield, and photosynthesis of the modified energy cascade crop model.
Hold in the schedule. That doesn't happen too often. All right, share screen, share screen. Here we go. Just screen. You'll get a peek at my lovely space farming wizard.
Definitely not AI generated. That looks real.
Oh no, it was super AI. UF has some thing that lets you make silly images, so I did. Anyways, my name is Donald Kuhn. I'm with the University of Florida Agriculture and Biological Engineering Department, working on the modified energy cascade model as part of my PhD here. And so without any further ado, I'll just get started since we're a little over time. Try to keep things going. We've talked about this. Everybody tends to know pretty well what it takes to have good production: optimum environment, non-limiting water and nutrients, and as little as possible pests and diseases. Do that and you'll have a beautiful crop. Don't do that, it doesn't look too good. And this is where you're going to start running into management issues, like how long can the crop tolerate these things, how much loss can be expected, what kind of effect would different interventions have, and most importantly, how does this impact your crop schedule for like the biological life support systems and keeping astronauts happy, healthy, and fed. If they're expecting to get food every 4 days and suddenly that's shifted, you're going to throw everything out of whack and have to readjust and get it back to schedule at some point. Now, how this is typically done is through the use of either really intelligent people with lots of intuition or a mathematical crop model. These models can take into consideration things like environmental conditions and crop characteristics, and all of our hard work in doing scientific exploration and statistical analysis, and combine it into equations to help process and make quantifiable predictions. Now, what I'm going to be talking about is the modified energy cascade models. This is a family of models. You can find the first one I'm going to show here, Cavazzoni's, in the NASA baseline values and assumptions document. And now keep in mind, it is the modified energy cascade. So there is actually the original energy cascade model developed by Bugbee, Volk, and Wheeler. And this is an addition to. So this one is parameterized for 9 different crops, predicts over an entire course of a day for the crop's life cycle, and it takes into account 26 different parameters to give you 13 different outputs, including total edible biomass and your transpiration rate. There are 2 other versions, though. You have another one by Boshiri, who really focused in and worked on it for closed loop systems, adding in extra considerations for gas and water flow, and then reducing it down to an hourly time step for more granular management. Only took a few more parameters to get a few more outputs. And then lastly, we have Dr. Amatranos, which went in the complete opposite direction and tried to simplify the original Cavazzoni one back down. And so I actually managed to cut out half of the different parameters, making it a much less complex model. But one of the things that none of these models did is a full evaluation looking backwards to see whether or not they performed, whether they added or subtracted uncertainty, sensitivity, and most importantly, accuracy compared to their predecessors, and especially in regards to the original energy cascade. Each one of them did analysis for themselves with their own data and their own crops. But like I said, they didn't quite go backwards, which is where I come in. And it's worth noting that the MECs are not full crop models that can predict everything. These are more a production potential model. So they work under optimal environments, defining the maximum production potential. There are things like water and nutrients that can limit that these models are not able to capture, along with any reductions from pests or diseases. It won't capture those. But if you're trying to figure out what is your production potential and then figure out what is missing from that, the MECs are pretty good. And so the goal of my PhD partially is to describe the effects of input variability and the model structure on the outputs of these MECs, and then work those considerations and these evaluations into the development and enhancement of this model for biological life support and controlled environments. And one of the ways I'm doing that is a global sensitivity and uncertainty analysis. And so this is following a Monte Carlo algorithm in which I look at the entire range of the model's inputs. So for the environmental ones, I'm looking from the environmental, from the operational minimum to the operational maximum. For the crop-specific parameters, I'm looking at the literature stated values plus or minus 20%. Within those distributions, I then select samples using the Sobel method, and I can generate a lot of samples across those ranges, and it requires about a few 100,000 simulations each. That way I'm able to perform the Sobel method analysis, which same guy, different thing we're talking about though, which allows us to break down and portion how much of the input's variation can explain the output in 3 different ways. The first order being if you just change temperature, how much of an effect that has on yield. If you change temperature and light, that'd be your second order where they're having an interaction. And then total order where it apportions the second order back into the first by just summing up the combinations there. And if you're thinking back and you're like, Donald, didn't you do this last year? Yes, but now with more. Now with more. And so last year, it was just the environmental inputs. This year, I've included the crop-specific parameters. And so the results have changed quite a bit. So you can see that the models still have a large dependence on PPFD as one of the model inputs over here. I hope you can see my mouse. Yeah, over here. With depending on the model version that you select between Amatronos and Cavazones, you can get about a 30% difference in sensitivity to PPFD. So for every micromole that you're adjusting on that, you'll get a 30% difference depending on which model version you're selecting. Otherwise, for many of the different parameters, they are very close, except for Cavazones over here, which has a very large dependence on the maximum value of carbon use efficiency in the model. And Bashiri's is very similar to Cavazones, except for one thing where it has all these interactions between light and different crop-specific parameters. But overall, Cavazones is the least sensitive model across all the different versions. Then likewise, looking into transpiration, we have slightly different looking charts here. Amatrono and Cavazones models behave very similar with sensitivities very close within 5% of each other. Bashiri's model, it behaves with much less sensitivity in everything except for this one strange crop-specific parameter here, which is actually an artifact of this model being developed hourly, but also for a closed-loop system that could be in space. DPG is the physical length of the day plus or minus 20% for this analysis. So just that slight variation in 24 hours plus or minus 20% means that it dictates about 85% of the model's transpiration rate outputs. That and about 2 or 3 dozen different interactive components between temperature and every other. Patrick, did you mute me?
I don't think so, but it was, yeah, you're back now. So,
Okay, it said the host muted me. I was like, oh, that's not good.
I might have touched something on the other computer. My bad. Please continue. I'm enthralled.
Okay. All right, this is where I am. Okay, so yeah, that's some of the Sobel analysis results. I'm working on that, but this is just model analysis. These are fake numbers, well, not fake numbers, but they're simulations. What about actually evaluating them and making sure that these models are correct? Because I don't want to just do a bunch of work and say, look at that. I want to make sure these things are actually working properly and do my own evaluations independent of model development. And so what I have going on, there's a series of growth chamber experiments that are utilizing 2 different varieties of lettuce. Waldman's green, which is what the model was originally parameterized for. And then the UF breeding ascension BG223-1251, which is a heat-resistant line of leaf lettuce being developed here at the University of Florida for controlled environments. The reason for this being is that one of the global sensitivity findings is that the model is rather unresponsive to temperature. So we want to include temperature into our trials. That way we can, bolster and improve the model a little bit. And then those are just some of the environmental conditions that we're going to be testing under. To capture everything we need, I have a small operation I like to call low throughput phenotyping with a growth chamber. I use a fluorometer to capture the photosynthetic characteristics, a light core for leaf level gas exchanges. I also have a Cyrus-4 connected to one of Dr. Melanie Carell's old hypobarrier chambers that I use for canopy scale gas conductances, and a small fleet of load cells to track daily transpiration rate and crop growth rate. And then in addition to this work, I also have news from the last Space Ecology Workshop. I was approached by Dr. Thomas Graham at the University of Guelph, who said he has something that he think would make life easier for me. And boy, was he right, because I really wanted that mass balanced growth chamber. That way I could get better information on canopy conductances and daily transpiration rates. And also that one thing I couldn't capture here at UF, which was daily carbon gain. So I'll be using those mass balanced growth chambers next year in addition to the work I'm doing here. And so what I do have is a little bit, I got sidetracked trying to prove myself wrong on this slide here. So in the orange and blue, you have the 2 different varieties, WG for Waldman's green and BG for that developmental line. And these are the weekly harvest values for dry edible biomass. And in the black line, you have the predictions for Cavazzoni. Now, this model technically has never been tested at 500 ppfd before. So this could be some of what we're seeing there, but I have checked and double checked to make sure that I didn't mix anything up. It was not that easy. So I did not make any mistakes here. I do believe that this model is not quite accurate at least these high light levels. Overall, though, the Bashiri version of the model does have very significant structural differences, from the others, including a large portion of interactions. Looking at PPFD levels, depending on which model version you select, you're going to get a very large difference in sensitivity. And these models are a little bit uncertain at the moment on whether or not they can predict lettuce yields at 500 BPFD. So we're going to be looking into that and double checking those results and probably fixing it. I already mentioned that a little bit, but quick acknowledgements to the undergraduates who help power my Arduino systems, some of the co-authors who are always pushing me to keep doing more and make it better, even though I think it's just going to be a quick thing. It never really is. Any questions?
All right. Yeah, thanks so much for that, Don. That was really cool to take some of the previous research and look at it with a closer degree of resolution. Sorry for accidentally muting you. I was controlling the wrong mouse. But yeah, I guess, are there any questions from the audience? I guess while we're waiting for them, I guess Jane would like to read some of the publications that you were using, if you've got a citation list, maybe.
Oh, yeah. These ones.
Well, there you go. Take a screenshot.
So these are all the papers from Kawazoni, Bashiri, Amatrano, Bugbee, Wheeler, and then a couple about the sensitivity analysis that I ran.
So now that you've taken a look at the modified energy cascade models and you think that they're not, I guess actually for calibration, is 500 ppfd common light level we see for controlled environment agriculture?
That's hard to say. People don't tend to like to tell you what their points are in farms. I know for experiments, it tends to be a little high. I originally requested something lower, and then I showed up and the plants were already in the chamber and I measured it higher. So I rolled with it because I was like, yeah, the model works there.
Except if it doesn't. Interesting.
Future analysis will also include the original energy cascade because Dr. Wheeler saw this presentation, not Wheeler, Bugbee saw this presentation a few weeks ago, and he thinks I should include that in many thoughts.
Okay. Are we going to be seeing a Don's version of the model next, or are you just going to be content with analyzing the other ones?
That depends on the results. I'm a firm believer in model development, not adding or making things more complex unless it's truly necessary. So if I can say they work good, this is how you use it, this is the range it works in, confirmed, I'll be happy.
Sure. Yeah, Christina wants to know if you're planning to do more growing plants in hyperbaric environments.
Sadly, I didn't do any of that, but if anybody wants to fund it, the pressure is a component of the MEC. So just get me a hyperbaric chamber and, well, a larger one, and I'll go for it.
Great. Well, we are on time now for the next portion of the talk. Thanks again so much, Don. That was really fascinating. We're on a coffee break. So we got 15 minutes, and then we're going to push through the last sprint, an hour and a half, 4 more, I guess actually 3 more presentations, because I just did Dr. Caswell's kind of out of order. So maybe we'll even get out of here a little bit early. So I guess, yeah, let's reconvene in 15 minutes and we'll hear from Ducky about his closed loop lab. Thanks, everybody. Talk with you shortly. Hey, Ducky, are you there?
Yes, Patrick.
Yes,
I'm here.
I'm here.
Great to hear from you. How you been?
I'm doing okay. A little frustrated today. I just had to completely rebuild my presentation.
Oh no.
The drive that was on took a nosedive during my drive to MDRS.
No way.
So I had to completely rebuild the picture presentation part of it. So it's a little more sparse than I wanted it to be, but at least I got all the pictures back. So that's good.
Yeah, that is good.
So I have a question for you. It looks like
Yeah.
we're just about time.
Mhm.
Do I have to have my video on to share screen?
No, you don't.
Good, because my connection is rather tenuous.
Sure. You
And
said
I
you're out
think
at
if I,
the Mars Desert Research Station?
yes,
That's
and I'm
fun.
using and I'm parked in the bus. I'm sitting in the bus, parked in the bus behind the outpost. So I'm actually about at the outer extent of the Wi-Fi reach.
Gotcha. Well, you sound pretty good, so I think you can go ahead and start whenever you're ready.
All righty, let's see if I can get the share to work. So we want that one. We're going to share sound and share. Let's see if that comes through. Says I'm screen sharing. Okay. Can you see? Yes. Cool. All right. Guess I dive right in, right, Patrick?
Yeah, go for it. I'm excited.
All right. A mobile closed loop ecological system laboratory, air, water, food, and waste recycling for one in a single lab. Closed loop life support is a holy grail of long-term space travel, but closing the many loops involved in providing fresh water, fresh air, varied food, and full nutrient and mineral recovery through waste recycling has proven daunting, to say the least. I guess I put the picture I didn't want up there. Sorry about that. Celebrating Halloween with family. That is the front of the Mobile Analog Space Habitat. We're celebrating Halloween. Okay, how do I do this? There we go. The Mobile Analog Space Habitat incorporates multiple species in a working ecological system in order to close these loops within the mobile lab. With two sources of protein, a vegetable garden, plus insects and cyanobacteria, we are producing air, we're recycling water, growing food, and processing our wastes on board the MASH. We are maintaining each population, and now we are working to engender reproduction within each of the populations while beginning to harvest from them in order to bring them to full. How the ecological flow works in this 300 square foot space. Module 1, energy. As you can see in the photo, the solar panels are actually in their upright position collecting energy. There are another 8 panels on the other side of the bus and 4 on the roof with a rated capacity of 6 kilowatts, although with their age, we're down to generating just shy of 3 kilowatts at the current time. A closed loop system must balance a myriad of factors that are shared between the modules, particularly energy, of course. Our power system has a rated capacity of 6K, but due to its age and so forth, we're producing between 2 and 3 kilowatts of power during the bulk of the sunlit part of the day. Now, here at MDRS, I've been blessed in that my batteries are very nearly fully charged by 9 a.m. and for the rest of the day, I'm operating on solar alone. So we're very blessed out here with the open sunshine. Each of the modules are designated very specific time periods that provide energy based on their specific needs. This means, for example, that in order for quail to lay eggs, they must receive 14 to 16 hours of strong light per day, while the marsh is allocated just 8 to 10 hours and the garden just 8 to 10. These periods are not designed to power produce, but rather to facilitate reasonably normal Earth-like growth within the confines of the mobile lab and using the available energy. In addition to lighting, there are pumps that lift water from the pond to the marsh and from the sump pump beneath the garden back to the pond. Also, there are 2 circulating fans currently running about 18 to 20 hours a day, the air pump that feeds air to the pond, and the photobioreactor. In addition, during the peak of our summer with exterior temps above 115 degrees in Phoenix, I've been running an evaporative cooler in order to keep temperatures inside below 95 degrees. And this is critical for the quail and has also benefited the plants in the gardens. As I have been living full-time in the MASH since January 2023, other energy requirements include running a refrigerator, stove, microwave, water heater, central heat, and mini split air conditioner. I have learned to balance the loads by choosing carefully when I task high power units such as the AC, washer, water heater, cooking, and clothes drying. My battery reserve, while substantial, is not endless. So managing the energy of the system requires a daily vigil. That being said, there are many habits I have developed to serve to optimize use when the high power components are necessary. The battery pack you see in the right is 24 6-volt deep size wired at a rated capacity 30 watts. That's just barely enough that I can operate for approximately 2.5 cloud.
Hey, Ducky, are you there? We're having trouble with your connection, I think. I guess that answers that question. Yeah, I think he did say that. I'm hoping we get to see some pictures of the inside. It's quite interesting. There's a lot of, well, I mean, it's a closed ecological system, kind of closed. Although, yeah, Ducky seems to have dropped from the call. He did say he was at the edge of the Wi-Fi range. Unfortunately, I don't think that he's published very much on it, Don.
Sorry about that, guys. I'm back.
There you go. No worries.
So where did it drop out at?
You were explaining to us your battery bank.
Okay. So we'll back up one. The battery banks, 24 6-volt batteries, series parallel, capacity 32,000 watts. Gives me about 2.5 days of cloudy day operation without sunlight. The air loop isn't just about using algae to consume the CO2 and release oxygen. It's also about having sufficient static air to produce the air bubbles in the pond and the photobioreactors themselves. Drawing room air that is used by the quail, the plants, as well, serves to close this air loop. The current single tube photobioreactor was initialized with a colony of spirulina in May of this year. These photos you're seeing, by the way, were taken this morning to give you an idea of their status. Now the colony's growth has exploded, and I am preparing, as you can see, I'm lining up the other tubes to split this first tube and begin our second tube's growth cycle. We'll continue splitting the tubes as they reach a strong density until there are 4 thriving photobioreactors. These units are equipped with a valve at the bottom to facilitate harvesting that will begin when the 4 units are sufficiently stable. At the current growth rate, I hope to have the 4 tubes operating by March of next year at full capacity and begin harvesting the spirulina for feed for use for the quail and the fish. In module 3, the water loop begins with the pond in the garden as the fish fertilize the water that is delivered to the marsh, depositing solids, then travels to the garden, less the solids, of course, carrying needed nutrients, then falls into the sump. From the sump, the bulk of the water is returned much cleaner to the pond, but a small portion is drawn off to dilute urine from the toilet and then is delivered to the solar distiller on the roof. The current distiller is a dual hemisphere glass unit with a 1 liter reservoir inside that receives our water to be processed. The unit takes about 6 days to process the 1 liter of pond urine water that is in there at this time. I have several other units under construction that will eventually improve production to reach a goal of generating 5 gallons of water, or 5 or 6 liters per water per day is the ultimate goal. It may take us a while to get there. As I bring the newer, better designed units online, I hope to increase this yield gradually. The UV light, there will be added a UV light and mineralization filter between the distiller and the collection bottle as we begin to prepare to integrate this into the drinking water tank in the floor of the bus. The marsh is a new addition this past year, providing the opportunity to grow blog plants. My apologies in the pictures because the drive down here, things were not as robust as I had hoped, and so we had a lot of sloshing. But the water lettuce in the right-hand picture and the rice in the left-hand picture actually survived, and the water lettuce actually is doing pretty good from where we started 2 days ago. Without the solids capture that the marsh provides, we would end up with heavy solids clogging up the garden and create other issues, aerobic development within the garden that would create additional complication as time goes on. As the marsh also provides a home to smaller minnows, guppies, amphibians, and catfish, small catfish, as well as a grow out of tilapia fry without the risks of the larger predatory fish. Of note is the fact that all of our gray water from the kitchen sink, shower, and clothes washing is delivered to the marsh on a periodic basis to then be filtered by the marsh, ultimately passing through the garden and then back into the fish pond. A design is in process to reduce our shower water use by actively recycling the shower water during bathing. Calculations are suggesting that a shower can operate with as little as 2 gallons of water using this recycling process. This is a picture of the entire farm. On the left, of course, is the fish pond. At the rear is the quail cage. Above that is the marsh. And to the right, of course, is the garden. You can see we do have pretty good growth there. We have corn in the foreground. To the right of the foreground are some bush beans, which I'm getting ready to harvest, a handful of pods. Behind the corn, I don't know if you can actually see it, is our tomato plant, which has just started to flower. And then down in the back behind there, we have some Brussels sprouts, a couple of lettuce plants, some arugula, and other assorted things like that that we're testing for growth. And as a side note, I discovered just yesterday that I actually have 3 flares of what looks like corn cobs beginning to grow. And we're not even at 2.5 feet height on our corn so far. So we're seeing definitely an improvement over the last crop I tried to do. The food loop includes the fish, the quail, the eggs the quail produce, the garden crops, and the marsh crops once we get those stable as food for us humans. The food is nurtured from the combined outputs of the very creatures we're also feeding. So too, the feed loop is drawn from the inedible discards of the kitchen and the garden in order to feed the black soldier fly larvae and worms, and the entrails, bones, eggshells from harvesting fish and quail, plus spirulina, duckweed, and moringa provide ingredients to produce feed for the fish and quail. We have been able to produce outputs from many plants, tomatoes, bush beans, lettuce, herbs, as well as Brussels sprouts, cabbage, carrots, and onions. Currently, the tests in progress are exploring the corn, rice, and water lettuce as well. And we have plans to include a fruit cocktail tree using espalier to train it against the white wall in the hopes that we can add fruit to the overall diet. Come on, there we go. The last module, number 5, is our waste processing, or what I prefer to call nutrient recovery. On the left, you can see our current prototype. I think we're up to 7 now of our black soldier fly habitat. This is an active toilet currently in use for a week now, and we expect to see the larvae begin to self-harvesting into the clear canister to the right within the next couple of days. On the right, of course, is our worm bin, which is still holding active after a week of establishment. We're very pleased that both are still running. In planning our waste processing cycle, the larvae have proven in multiple papers, as well as what we're finding here, to be a pathogen, basically, wall. The larvae process the human waste, the quail waste, as well as kitchen waste and others. And the compost that is delivered and harvested from the toilet bin then is moved into the worm bin, which we have learned and discovered on our own, as well as from papers, that the worms treat the compost from black soldier fly as a delicacy and are voracious to consume it. In the process of consuming the compost from the black soldier fly, we are blessed that we are delivered potting soil that we can then use to expand our garden. This gives us a route to be able to actively improve the garden on the moon and Mars by simply impregnating the regolith with the potting soil and all of its biological materials when we arrive. We have very limited automation in our system. In fact, this is our automation panel, as it were. The lights are on timers for the specific needs of each of the various species. The garden sump is activated by a float as it fills, as the garden itself drains. Currently, I manually stir the algae each morning, and I add pond water every morning as feedstock for the algae. The growth that you saw in the algae tube is just 3 months growth. As well, I'm manually feeding the fish. I do have an automated fish feeder, but I'm finding I like feeding the fish once a day every morning. And I'm finding these actions of caring for the species has a very pleasant and positive aspect of my daily life here in the MASH. Our future goals for the coming year include integration of the system modules to finalize the photobioreactors and begin harvesting the spirulina as feedstock and document the oxygen recovery at that point. 2, to finalize the distillery and hopefully bring our capacity up to 1 or 2 gallons by March or April. Begin a higher density planting of the crops. We're only using about a quarter of the garden bed at this time, and we're hoping to double that by January and get to full capacity by April or May. The beauty of the planting within the MASH is that I do not have to deal with seasons, as I have climate control within that keeps our ambient temperature in the range between 74 degrees and 95 degrees. And lastly, we're hoping to finalize the toilet between 1 or 2 more modifications, and then be able to begin harvesting larvae for our feed stock. Thank you for listening to me. We are on the web at earthseed.space. You can reach me directly through the doc at earthseed.space, and we're also on LinkedIn and Facebook. As a 501c3 nonprofit research organization, I've reached the point now where we're beginning to look for board members, as well as begin moving forward into our phase 4 of our business plan. I'm in the process of acquiring land to the west of Phoenix, where we will be constructing a full-size 7 to 10 person closed habitat at that location in the next coming 2 or 3 years. Thank you very much for your time. Any questions?
Wow. Thanks so much, Ducky. That was really cool. I'm sorry that we didn't get to see your previous presentation. I'm sure it was even more fantastic.
Okay,
Yeah,
how do I turn off?
I think that's the first time I've seen any of the inside of your MASH vehicle there. We have just a couple of minutes for questions. Kevin wants to know what percentage of your food comes from the MASH.
Very little, as right now we're experimenting to identify the crops that will be successful within the MASH. Again, we started with corn this time. Out of 12 plants, I actually have 2 that have survived this far, and the one is actually generating cob husks at this time. So we're going to call that one a success. So our next crop will include corn. We'll also be including rice, as well as the lettuces. I hope by March to be able to harvest at least 2, maybe even 3 meals out of the garden, at least a week. I'm not sure of what our yields are actually going to be. We know we can get quite a bit, but that's what the hope is.
Mm-hmm. Okay. I think that since we're not going to have our last presentation that's on the schedule, I'm going to go ahead and push 5 minutes into the next one for a little bit more Q&A, because I'm also really curious. Frida wants to know what you do with the adult black soldier fly larva.
The adult black soldier fly, there is a tube that goes from the clear chamber back into the feeding chamber that allows the adult flies to return to the feeding area. In the ceiling of that waste area, there is a honeycomb piece where they will then lay their eggs. At that point, the flies, after mating and laying eggs, they typically live only 3 days and then die and become part of the food chain themselves. The life cycle is kind of interesting in that regard. And then the only thing that we need to, when we do our harvesting, which is the lid on the clear chamber, we are then able to harvest any adult flies that are left plus larvae as part of our feedstock for the quail and the fish.
Okay. Yeah, we didn't get too much detail on the quail in this. Tell us more about the birds.
The quail are jumbo Coturnix quail. When I have a full covey, this is the last bird of the current covey. I'll be harvesting her probably at the end of the month. The Coturnix quail will deliver a 6 to 7 ounce chunk of meat on harvest. They will deliver approximately 1 egg per day for about 320 days. The plan is that we will maintain an active covey for approximately 1 year at a time, and then we'll have a secondary covey in a second cage that will produce meat on an 8-week cycle. So we'll be able to harvest at least 8 quail every 2 weeks.
Wow.
And the tilapia we just had our second crop of tilapia born about a month ago. We started with 20 tilapia fry 3 months ago. We now have a little over 75, and we'll be harvesting the original 20 in the next couple of weeks with 2 fillets per fish of about 7 to 10 ounces each.
Oh, okay. Those are getting pretty big then.
Yeah, they're almost a pound each.
Wow. Yeah.
Already.
How
that's just
big
in
is
3
the
months.
aquarium they're in?
We had a lot of sloshing, so I'm down to about 150 gallons of water in the tank. And like I say, I've got about a little over 75 fish, so I'm at half capacity of fish.
Okay. I think I recall when we talked about this previously, you mentioned you were putting the wetland water into, or maybe the fish water into your bioreactors to top them off. Are you sterilizing that first? Because I feel like you could pretty quickly introduce algae predators.
That is true. Algae predators could, but there are several natural barriers that reduce the number of creatures that enter into the MASH that I'm not ready for. I do have a handful of spiders who came in with the mosquitoes. But for the most part, I do have an insect light in the back, a UV bug killer, and the minnows and the other small fish in the marsh feed on those mosquitoes and larvae and things to keep the marsh clean. From that, I also have I forget what they call the fish. They usually stick to the sidewall and they eat the algae. So any algae that grows in the marsh is also consumed by the smaller fish in the marsh. Somebody asked about how did I develop the idea for the project.